Yes, please speak.
Hello, everyone. It's our greatest privilege to have Dr. Evnimeluch Erum, Professor, Head of Civil and Engineer, Environmental Engineer Technician, Israel Institute of Technology. He is one of the chief architect and founder of Bifloc Technology. He has worked in various capacities in different organizations, namely Chief Scientist, Israel Ministry of the Environment, Dean, Department of Agriculture, Agricultural Engineering, and Director, World Aquaculture Society, and many more. He is very happy about the Indian government's move of Pradhan Mantri Matsya Sampada Yojana with a budget of rupees 20,500 crores for augmenting fish production in the country. Sir, we request you to kindly deliver the keynote. Thank you. World Aquaculture Society and many more. He is very happy about the Indian government's move of Pradhan Mantri Matsya Sampada Yojana with a budget of rupees 20,500 crores for augmenting fish production in the country. Sir, we request you to kindly deliver the keynote. Thank you. World Aquaculture Society and many more. He is very happy about the Indian government's
Good morning, everyone. Do you hear me? Do you hear my?
Yes, yes.
Yeah. Okay. Good morning, everyone. It is really my pleasure to visit with you. I have been in India many times. Do you hear my? Know a lot of people,
Yes.
but visit for all that period of the pandemic. Good morning. Hope that in a few months we can meet in person. It is really my pleasure to visit with you. I have been. I will try to speak slowly so the echo disturb us. What we are going to do next half an hour or so, I would like to give you a very brief introduction and then a few possibilities for the future, how to get biofloc technology to help Indian aquaculture. I need to larger so I can control it. Okay. No, that's not. This is not it. Dr. Shivakumar, can I get the control of the PowerPoint? No, this is not the one that I got. I want to say something. Usually conferences like this take a few months to organize. I have to say that Dr. Shivakumar and his department did everything in less than 1 week, and it's wonderful how it was organized, but there are when you do it so quickly, there are some problems. I'll try to follow this lecture, even this is not the one I planned. So we want to get in our ponds to have high production. For the high production, we have to put a lot of feed. A lot of the feed residues are left in the pond, in the water and the sediment, and we have a very high load of nutrients. I cannot move it. Okay, this is my slide. My presentation. Okay, here we are. Okay, that's in the pond, and we'll move forward when we have biofloc technology ponds. One of the principles, no water exchange or very little water exchange. This has a few implications. One, we do not waste water. Due to evaporation, we lose between 0 and 4% of the water per day, very little. We can even produce fish or shrimp in the desert. You have in India a lot of dry lands. You can use these dry lands to produce fish, and it can be very successful, very good way to use these areas to give some income to farmers in these areas. Another implication of the minimal water exchange, you don't introduce polluted water, you have a perfect biosecurity system. When you don't exchange water, the feed and the feed residues stay in the pond. A lot of feed is accumulated. This is accumulated in the water. It is a problem, but it is also a big advantage. One advantage or one implication is that with the high amount of residues in the water, high amount of organic matter, nutrients in the water, a lot of bacteria can develop. The concentration of bacteria in biofloc system about 10 lakh to 100 crore per milliliter, a huge amount, 1 million to 1 billion bacteria in 1 small milliliter of water. Another point, another advantage of the minimal water exchange is that you don't pollute the environment. You discharge very little water, so you don't pollute the environment. Very important, very needed, and laws like, and you were some of the pioneers in the world at developing laws preventing pollution of the environment. Is my voice going through? Do you hear my lecture, Dr. Shiva Kumar? Okay. Now, when you have a lot of bacteria, high density of bacteria in the water, the bacteria aggregate, they join together. And this is sort of a cartoon showing that the bacteria stick together and they have company and they are very happy. But this is a cartoon. Now we can look at this biofloc, aggregate of microorganisms. And this is an electron microscope picture. And you can see here filamentous bacteria, larger organisms, different organisms, a huge community of organisms. But it is part of aggregate, the biofloc is open, can get through, oxygen can reach bacteria, nutrients are moving, so physically it's a very good system. Another picture, but this now the picture taken by an optical microscope, low magnification, and the smaller dots, this is a biomass of bacteria. Here you can see larger organisms. These are, in this case, protozoa that come to eat the bacteria. There are nematodes, zooplankton. The biofloc is a community of a high variety of organisms. In a biofloc system, you have about 1,000 species of organisms, and it's very important for nutrition, as we'll mention, very important for the immunity against the diseases. So this is two pictures of bioflocs, and we move forward. I want to say that in the present lecture, we'll get very, very brief introduction of the biofloc. Later on, we'll discuss the ways to get more knowledge and more practical knowledge, more information, how to run your own biofloc farm and to make profit. Now, having a lot of bacteria, there are good and bad points of view. Now, if you do not control the bacteria, you have a lot of bacteria not controlled, they will kill your fish and shrimp. You have to control bacteria. Needed. There are chemical ways to control it, and you can use the bacteria to be very, very beneficial. Now, one way to control the bacteria is adjust the feed. You want to have feed that have a proper ratio. Usually we add feed that has a lot of proteins. The only part of protein is utilized. A lot of ammonia is excreted. It can kill your fish, but you can control it by adding feed that contains a lot of carbon. Usually take the one. So one of the possibilities is to use molasses. It's easy to add the molasses. Usually they're not expensive, but you can have rice bran, corn meal, wheat meal, a lot of other components in different places. The prices are different, and I recommend to use different components. And if you control the bacteria, they help you very much in producing high crop of fish, high crop of shrimp, healthy and nutritive. Let us move on and see some other aspects. Now, when you have a lot of bacteria, as we said, 1 million to 1 billion bacteria per milliliter. This is more bacteria or as you find in most biotechnological industry. You have in the pond, you have a biochemical factory, very, very active. Now, the bacteria eat all the waste or organic matter. And then they degrade it. You don't accure most of the organic matter. They also degrade the toxic materials that are in the waste. You have to realize that if you have high density of fish, no exchange of water, all the excretion, all the shit of the bacteria is in the water. You don't want it to stay in this way, so the bacteria degrade it and develop new bacterial cells. So by controlling carbon to nitrogen ratio, aerating the pond, mixing the pond, we can have what high quality. The bacteria are the no toxin, and the fish can grow there well, and we'll see some other advantages of this system. I hope that you hear what I'm saying. So please note with your hand if not working. The flock can be used as feed. Now we have to realize that most of the feeds that we give to the pond, and it's expensive. The biggest expense in growing fish or shrimp is putting feed pellets in the water. 5% of this feed is accumulated into the body of the fish, and you get bigger and bigger fish that get to the market. 25% is used. 75% is excreted into the water. Usually in conventional aquaculture, you don't use this one, what was excreted into the water. It settles down, you have to change the water to get it out, you waste a lot of money, you pollute the environment, and your fish are not growing as they could. Now, due to the fact that these organisms are found in water as bioflocs, the bioflocs have a size of about 1, 2, 3 millimeters. So the fish or the shrimp can fill these, can eat it, and it's a very nutritive. So actually, when we have a biofloc system, the fish or the shrimp eat twice. First, they get the feed pellets, they eat the feed pellets, use 25%, excrete the rest. The bacteria take it from the water, form bacterial cell, form microbial protein, form the bioflocs, and the fish eat it again. So the fish eat twice as compared to the conventional system, that whatever was not eaten by the fish when he eat the feed pellets is lost. Here we recycle the feed. And feed utilization in the bio pond is roughly 2 times higher, twice as much as in conventional ponds. So You use the feed very well. You save on buying feed. Again, it's the most expensive operation, the most expensive expense in production. You use it in a very efficient way. Moreover, your fish eat two kinds of feed. One is the feed pellets, the synthetic chemical feeds that was produced in the factory. The second is the biofloc. The biofloc was not produced in the family. The biofloc is a natural feed produced by Mother Nature, containing a lot of components that are not found in the industrial feed, and enable the fish, enable the shrimp to be more healthy, to grow better, and to have higher resistance, higher immunity against disease. Now, this is a picture taken in Israel of floc system with red tilapia. You can see how many fish we have in the water. And all these fish grow, are healthy, and you get very high production. At least twice as high conventional systems. Sometimes it can be 10 times higher than in conventional systems. Again, you will not get 10 times higher in the first cycle of production. Later on, when you get more proficient, when you learn to do it, you will get better production. Bioflocs are also the best medicine. One of the big in aquaculture is disease outbreaks in shrimp and fish production. And it can be horrible. If a viral disease gets into a shrimp pond, within a few days, you can have all the fish dying. You lose everything. And it happens in many countries. Again, I have to compliment the Indian aquaculture. You found the way how to take care of diseases, and therefore you became the first exporters of shrimp in the world. With biofloc, you have systems that is and it was found recently, there are a lot of research works with tilapia. There is a disease that is caused by streptococcus. When biofloc, this disease is lower. It's not such a big problem with the fish. And again, I did when I was working in Africa in the farms that almost lost everything because of white spot. We started to change it to system. This farm is now productive and profitable and growing. So there are a lot of possibilities. The use of bioflocs prevent not to zero, but to minimize the disease problem. And different kinds of ponds. Here, this is you have here small, relatively small ponds built out of plastic, very easy to assemble. There are different sizes, different producers, different ways to do it. I'll not talk about it today because we cannot talk about everything in a very brief introductory system, but there are some things that have to be paid, really done very carefully, to have good slopes, to have good drainage, to have good aeration. I will show you something about aeration in one of the coming slides. Uh, aeration, oxygen, aeration, and sludge. Uh, I've been visiting in my work in farms all over the world. I've been visiting many, many farms. Some of them are very successful, very productive, very profitable. Some of them failed, and people lose their crops, lose their money, and they say, okay, it's not going, it's not working. The main reason for failures of aquaculture ponds is through an improper aeration and improper control of sludge. So it's very essential, very important. And again, I'll mention it. We'll not go into the details, but you have to keep the pond oxygen. You have to provide aeration when we have high biomass, high fish or shrimp biomass in the pond. With the bacteria, they also need oxygen. You have to provide enough oxygen so that oxygen is above the critical level. I'll not give numbers, it's different for different species. It is different in fresh water or in salty water, but later on, when we'll continue with the work on bioflocs, we'll get to the details. You have to mix the water. The water has to be properly mixed. You have to minimize sludge accumulation, organic matter that settles to the bottom, accumulate at the bottom. If the bottom is not aerated, in most ponds, the bottom is not aerated, it becomes anaerobic with no oxygen. It gets to be rotten, it gets to be black, very bad smell, and a lot of toxic material. This is one of the enemies of the farmer. You have to control the sludge. It's very, very, very essential. Uh, if you don't do this, is how your fish would like look. You have to do all of this aeration, mixing properly. Otherwise, your fails. Uh, I would say the most popular, I don't think that it is the best. It has a lot of advantages, of disadvantages, a lot of problems, but the most popular aeration system in large ponds is the pedal wheel aerator. Pedal wheel aerator splashes water, small drops of water into the air. The drops of water that are in the air are in contact with the air, they absorb oxygen, get the oxygen back to the water, and in the same time get CO2 out of the water into the air. So it's a very popular system, very popular instrument. We'll talk, it's not the best one, and it has a number of disadvantages. One of the very common aeration system, and if you go into small farms, family farms, small individual farms, the most shown here in this picture, and this is a short, I'll show you a short video. You put airstone in the bottom of the pond, or you put tubes, perforated tubes connected to a blower that release small air bubbles. And here you can see here and here, these are the air bubbles that are going up from the bottom up to the surface. Usually fish ponds are water depths of about there. It takes a few seconds and these bubbles that were released from the air stone up out of the water part, only small part of the oxygen. So they are not very effective in oxygenation. The other problem with this system, when you aerate the pond with this, you do not control movement of water. Knowing this, I developed something, I don't know, I don't see myself. Uh, I developed a new aerator. I call this the Omega aerator. And again, I will not go into the details. It is also based on passing air bubbles, but passing air bubbles through I did it a few months ago. There was a farm in the Dominican Republic. They grew tilapia in earthen ponds. You can see the earthen ponds. Ponds were dug in the earth, covered with plastic, polyethylene. This is one of the cheap way of doing pond, having ponds. The size of each of these ponds is 80 square meters. And they had problems and they brought to me. I couldn't come to the Dominican Republic. It was a little bit before the pandemic. Later on, it was impossible to move. But I had internet connection with these farmers, and slowly I taught them how to make themselves the better aerator. I will show you now the video, and the video shows the aeration in the control ponds, in the ponds with the stone aeration, the ones that they used before, and the aeration in the ponds with the new aerator, the Omega aerator. Look, when you see it, you please see it, that in the control ponds like this, there is no water movement, and in the ponds with the Omega new aerators, you will see how the water moves. So let's go to the video. These are the control ponds with the airstone system. Now we go to the pond with the Omega. These are the aerators here. See how water moves all around the pond. Water is mixed, and most of the water flow, water flow is on the pond bottom, because the aerator get the water from the bottom, move the water through Omega with long contact with the air bubbles, absorb a lot of oxygen, and release the oxygenated water at the bottom. So I'll try to see it. We'll move it back. We'll try to see again. No, work. See the control, the one with the angles, and we go to the. Now this is the one with the Omega, and again they made themselves in the pond. And water is mixed, very good production, very high water quality. Production is going up. The farmers in this farm in the Dominican Republic are very, very happy with it. Now, we'll mention the possibility of continuing our contacts between Dr. Shivakumar, myself, others, working with people in India, with farmers, with leaders of the farming community in India, teaching them good way to control biofloc systems. One of the things that I will teach you is to produce yourself in the farm very cheaply to produce these aerators, get aerations that takes less electricity. In this farm in the Dominican Republic, they don't have electrical supply. They have photocells, they produce their own electricity, very limited. So for them, it's very important to use the oxygen to aerate and to mix the ponds with very little energy. So we'll go into it in details later on. We'll hopefully continue our contacts. Just to show you, this is a pond, a biofloc pond in the United States. It's a round pond, aerated with pedal wheels. The size of each pond is about 1,000 square meters, which is quite a bit higher than the average size of ponds in India. By the way, in some farms that I visited in India, they have very small plastic ponds. Too small is also not economical. Again, plastic ponds, this one is in Mexico. You see the ponds, very simple to construct. And here, this is the aerators that is aerating the pond and mixing. It's an Aero 2 aerator, not very popular in India. It can be quite good in a pond like this. I want to get to the summary and then I'll turn the presentation to Dr. Shiva Kumar Magda, and he will see if we can have question, discussion, or whatever. A few basic points that I want to stress here and to stress as much as possible. Biofloc ponds can help you produce more shrimp, more fish. I wrote here about twice as much per unit area, per unit water as before. So you can, and in India, and the Blue Revolution in India, planned to increase for the well-being of the farmers and the well-being of the country. Biofloc system can help in achieving this goal. Today, it's not too expensive. Biofloc system, you can start it small. You can start it with a little bit lower, less production than the maximum. A small farmer, a family farm can follow this with reasonable investment and get much higher income. So it's both biologically, economically, it is a feasible way to increase the production. You can grow fish in dry area. We mentioned dry areas in India, you can use, you grow fish, shrimp. You can save feed, reduce disease, and make a profit. Yet, I have been in places where they showed me a pond that said, here, we have a biofloc pond. So it was a pond with dirty water. Not every pond with dirty water is a good biofloc. It's complicated, really. It's the biofloc system is a very friendly system. You can do mistakes, but you can correct it. Here we have a biofloc pond. So it was fairly well. Water. I hear myself twice now. but this is my last sentence, and then we can see if this is the end or if we have some discussion. I talked briefly and we corresponded briefly, Dr. Shivakumar and myself. I did it before, tried to convince people that we have to find a good way to train people in India how to work properly with biofloc system. I cannot go into details. We'll have, but we groups of people that will first learn the theory, and they will have a pond. I promise to help them with all the instructions. Hopefully, in a few months, I can come and visit India. I'll see you in person, and we will with the Dr. Shiva Kumar and other experts in India. We can train farmers to do biofloc properly, and this will help achieve the blue revolution. With this, I would like to thank you for your attention. I hope that you heard what I am saying. And Dr. Shivakumar, I don't know if there is a place for question, discussion, else. Uh, you take over from here onward, please.
Yes, Dr. Arun, thanks for an excellent presentation, sir. Thanks in turns for sharing your knowledge and creating more interest in taking up the biofloc technology successfully. Thank you very much, sir.
Questions.
It was my pleasure. Should I get out of the system now?
So kindly do stay on.
Okay, I'll
Dear
stay on.
participants, kindly do reserve your questions and it will be answered at the end of the session. Kindly do cooperate with us. I now request Dr. Babita Rani, Senior Scientist, ICRC Mumbai, to kindly present. Thank you.
I can press stop share.
Unmute Dr. Babita Rani, please. Babita. Dr. Babita?
Yeah, I unmuted.
Please, yes.
Actually, I was not getting the unmute control. I'm sorry.
Okay.
Yeah. I would like to share the screen now. Sir, is it okay?
Yeah, please.
Sir, please enable me to share the screen.
Make a host. Yes, you're enabled, Dr. Babita. Now we can share the slides from your system.
One minute, sir. Yeah. Screen is visible, sir?
Yeah.
Yeah. Good morning, everybody. I'm really grateful and honored to be part of this program along with father of biofloc technology, Dr. Yaram Avni Mele. I'm grateful to the organizers, especially Dr. Shivakumar Magda sir, for giving me this opportunity. I'm going to talk about biofloc technology and its prospects in India. So,
Dr. Babita, can you increase the sound please in your system?
yeah, increase. Is it audible now?
Yes, yes.
Yeah, Indian aquaculture sector is performing comparatively very well. It is contributing to almost 6% of global fish production. And it is, of course, the recent days, we have per capita consumption enhancement also visible due to the awareness regarding healthy fish as a healthy food. At the same time, we are also facing challenges in terms of shrinking land and water resources. And we also have a lot of demand and supply gap in terms of feeding the population with quality protein. And when we want to face this demand supply gap to be closed, then we have other challenges like increased cost of production for feed protein to sustain the fish. And other issues like biosecurity issues we are facing a lot in almost all the species which we are culturing right now, and environmental sustainability issues through discharge of effluents into the land. So these challenges are also creating much serious setback in front of us in terms of generation of lot of waste and pollutants if we want to think about farm intensification. So how to control this waste and pollutants? We were researching and we were also thinking about many sustainable alternatives to address this issue. And also, when we think about farm intensification in Indian scenario, we always have a problem of availing cheaper rate and availing cheaper rate quality protein, like which can be affordable by a small or marginal farmers to continue their culture activities. We have a huge present percentage of small and marginal land holdings and people who are interested to enter into fish farming. So that is the real interest in if we think about Indian scenario in this biofloc technology. So when we talk about farm intensification, the sustainable alternatives only we can afford because we can't afford further pollution into the environment. Since we have already stressed out our most of the water bodies with pollution load, so we cannot any further pollution through farm intensification is not justifiable. So we have to see always sustainable alternative which we can do at our backyard scale or for which can be also useful for the small and marginal farmers. So this is the criteria when we think about farm intensification. In this case, in this situation, this biofloc technology is providing us a very good, very good, very good practical way, sustainable practical way for for the farmers of Indian aquaculture. So by biofloc technology, we talk about already. Dr. Yoram sir had briefed about what are the important benefits of the system. It can be intensive to super intensive production. We can also increase the productivity and reduced water consumption in terms of minimal or zero water discharge system. Biosecurity is ensured because we are not doing much of water exchange, and pathogen introduction and pathogen load is controlled in the system. And this system, the biofloc is nothing but protein, microbial protein. It is almost 32 to 38% protein. Many researchers have worked out. So it's full of protein. So we don't have that problem with lack of protein. I mean, most costly affair in our farm feed. So this particular protein is available throughout in the water itself, in situ protein. So here, how we are making with the help of exploiting the heterotrophic pathway by modifying the carbon nitrogen ratio of this biofloc system. Nitrogen is there in the form of feed waste and fecal matter. We are adding carbon, external carbon source which is available at our locality. In India, Indian agroclimatic conditions, we have a lot many sort of carbon sources which can be considered, which are having 50% and more protein, carbohydrate can be utilized for as a carbon source. So such resources are available at various agroclimatic regions in the form of rice bran, wheat flour, cassava meal, sorghum meal, even tapioca flour and jaggery and molasses, whichever is available at affordable rate in our area, we can use it. So this is how the biofloc, we already have seen some of the pictures of biofloc, and this is a microscopic and electron microscopic picture. As Dr. Yaram had already said, it not only contains only bacteria, it contains other organisms also. Going ahead, we had conducted some demonstration program in Kerala. We have along with government of Kerala. So we have here done the traditional method as proposed by Dr. Avni Mele. This we are using the pond soil and inoculating it along with ammonium fertilizer and fermented. We were using jaggery as carbon source as it's available and in plenty and at a cheaper rate. So this is a method we used, and the tanks were, of course, we are aiming at backyard scale farming, small and marginal farmers. So we were smaller tank. We were going with 20,000 liter, 20-meter cube tanks. And Indian conditions, farmers are mainly doing at the backyard scale. They are going from 10 to 50-meter cube tanks, starting from 10-meter cube. But of course, for better productivity, we can go for medium and bigger tanks. So then again, materials, any type of material which is having proper water holding capacity, we can go for wire mesh, GI sheet, or any such thing which is again locally available, which we can make it, we can use it. And along with this capacity varies from 4-meter dia to 5-meter dia, even more than that, up to 100-meter cube tanks already constructed by many parts of India. In many parts of India, and only in many important prerequisites is sludge management. We are finding here sludge management. One pipe you are seeing here. This is like on central drainage pipe. It is mandatory for every biofloc system. If it is small or big, we should have a proper slope towards this drainage system, so that the sludge removal will be proper. This is a prerequisite. Dr. Ramsar already told about aeration, so I'm not going into detail again about aeration. So this sludge removal, as and when required, we have to remove the sludge, and sludge control will save many of the problems, will solve many of the problems which are faced by biofloc farming. And in our demo units, we have faced challenges like I have listed here. One, slow alkalinity. This is a serious issue in many parts of India, wherever high rainfall is there. So such areas, I mean, very low alkalinity. Low alkalinity, it starts with like even 25, even less than that. That is not suitable for biofloc system. So many places we have faced these challenges in Kerala especially. So this alkalinity need to be improved for sustaining the biofloc in the long run. Then acidic waters. We have to check these water quality parameters before going into biofloc system. This is a suggestion. And in between also, this parameter we have to check for sure to avoid any chronic stress in the system. Then higher sunlight. In many parts of India, we are a tropical climate, so many parts we have this higher sunlight will create our heterotrophic pathway into photoautotrophic pathway. So suddenly it will shift to green floc, and farmers may face problem in nitrogen ammonia accumulations from accumulation of nitrogen or ammonia. So this we have to avoid with the help of proper shading. Wherever heavy rain is there, it's better to have a rain shade also, and then a proper light cutting And it's a prerequisite, like 60% light need to be reduced. Sunlight need to be reduced so that heterotrophic pathway will be promoted. And maintaining CN ratio, carbon nitrogen ratio maintenance is another prerequisite. There are formulae available. There is not much time to discuss that in detail. So when we stock the once we stock the fish, we have to maintain the carbon nitrogen ratio. There are various methods, calculations and all available online in many references. Uh, this is the in steps when we do the biofloc. When we inoculate it, we have prepared an inoculum. After inoculating it, gradually it will become green color, but it will transform. The transformation phase is like foam formation will happen, and then gradually it will become brown in color. So that is the transition phase, and then there is a floc formation, proper floc formation. So to make this, maintain this, whenever we are finding ammonia as like more than 1 ppm, so we can go with carbon nitrogen ratio, CN ratio of 10:1 to 20:1, depending on how much ammonia is there and which is the stage of your culture. Initial stages, we will keep higher carbon nitrogen ratio. Later on, we can reduce it to a maintenance ratio. We have in our unit, we have stocked once the floc reached 5 ml floc volume, then only we have stocked the fishes. And one important thing is that stocking the healthy seed. We can't afford a loss in seed quality, because in major pockets, many pockets of India, we are facing the problem with healthy seed availability. We have to see the seed is certified, or else we have to take the risk of getting it certified from authorized laboratory. It's always better to stock the certified seed. And when we stock, we should have an aim, like what should be our production per meter cube. So we can aim at anything between 25 to 40 kg per meter cube in 1 meter cube. So when as a beginner, it's always better to start with a lower density and gradually improving. That is a suggestion always we give to the farmer. Don't jump into very high stocking density and end up in loss. So the thing is that it's a nice technology, wonderful technology. We have to set our hand on our own. At a small system, set our hand, then escalate into a better system. That is the important thing, so that now it's tough when many things will come, many parameters are involved in this maintaining this biofloc as in proper suspension. Aeration is the main criteria. Then other water parameters, like I already mentioned, pH and alkalinity also creates a proper issue in the system. So we have to maintain all these parameters. Then coming to species, in India, commonly cultured species, tilapia, pangasius, in some pockets, carps are cultured, but carps don't tolerate higher density. That's a real issue. Smaller or minor or medium carps can be an alternative in this case, but my harvest size, in the final harvest size, is smaller compared to other Indian major carps. Then koi is very popular in the northern side. Shrimp is picking up in many parts of India. Magur and Singhi is also very popular species, which are among the northern pockets of India. In case of tilapia, we should specially mention that only improved strains are allowed by rule. Gift tilapia, monosex Nile tilapia, red tilapia. These are the improved strains which are only allowed, and not the other strains of tilapia. So whenever we are selecting a species, the first and foremost thing is consumer acceptance and marketability and the local area. Local market should be surveyed first. Then selecting the species, whether the species can consume floc or not. Because the floc is very rich in protein. If the species is not consuming floc, it is like we are not able to utilize the full potential of the system. Then high density tolerance. This is how many times carp fails, other species wins in this. Then disease resistance. We already mentioned disease and biosecurity is very important in aquaculture. So disease resistant strain or species are in that case, the indigenous strains are comparatively very good in disease resistance. These are some of the popular species I have just mentioned already. When we talk about the prospects, I will surely mention some of the research outcome which we have already done at various parts. From 2013, we were doing in this line. So some of the projects we have done, and of which right now one project is ongoing with World Bank funded project. A part of it is on biofloc. So we are conducting this biofloc based farming for inland saline also. We have studied species like Vanami, Rohu, Fungaceous, common carp, various tilapia strains, and Amur carp. We found these benefits like higher growth and stress amelioration, higher immunity, higher disease resistance, better carcass quality, higher digestion and nutrient utilization. And we have also done some broodstock quality in case of Cyprinus carpio, common carp. We have found brooder quality is also good, comparatively good in the system. These are some of the water quality dynamics parameters we have studied, but at the same time, this is just for information. Ammonia is a major parameter which we need to always control, which we need to control. Less than 2 is always better. Nil is always better, but it will go accumulate during this time. But if it is more than 4 ppm, we experienced mortality. But within that, we can control with the help of carbon source. We can, even sludge removal can control this ammonia. Nitrite also up to 6 ppm. Many researchers have identified up to 6 ppm, there is no problem. Nitrate also up to 60 ppm, people have found it's fine. Alkalinity, anything above 100 ppm is always better, and it should not be much lower. These are some of the other findings, like we can considerably reduce the feeding rate in protein as in situ protein is there, in situ feed is there. So these are some of the benefits of the system. This is from our own system. We have understood 32% protein is there in one of the biofloc unit, in the biofloc. Then we can also, best recycling is happening in our system. With ammonia is assimilated to form the biofloc. As Dr. Yoram had already said, we can make biofloc as a product. We have made it as a dry powder, and we have used it as a feed ingredient, and we have made a feed also. Okay. So this is also giving us an avenue open for using the waste into wealth. These are some of the more scientific responses. Scientifically, we have evaluated responses in terms of, we got higher immunity. Here we can see the conventional system, and these are all biofloc of different carbon sources. This is just an example. In this control is normal system, and this is these are all biofloc system. Here we have challenged the fishes, biofloc reared fishes and conventionally reared fishes with Aeromonas hydrophila, and we found a relative survival, percentage survival, quite high in biofloc reared fishes. This is a clear indication of disease resistance and higher immunity in biofloc reared fishes compared to conventional system. And the same is also observed in case of nutrient utilization. This is also protease activity as an example. Digestive enzymes would give good activity in biofloc reared fishes compared to conventional fishes, conventionally reared fishes. So this is these are all some of the glimpses of our research work to convince you that this is a one of the best system in terms of getting higher immunity and digestion. This is another activity we have done where we understood that, of course, biofloc shows your dirty water, and the consumer will definitely see that dirty water fish will be appealing, appealing or not to the consumer. So we have done sensory analysis with both cooked and raw fillet of this red tilapia, and found that biofloc reared fishes have much better sensory attributes and good quality carcass. So that also indicates that it's giving a positive impact on off flavor reduction in tilapia or any other species. We can also see that in biofloc we are getting a better response in terms of carcass quality. Another avenue in front of us is rearing the brooders in biofloc. Of course, we have got a preliminary result in terms of better broodstock quality and better, even we have done for common carp, we have got better broodstock quality, higher survival of larvae, everything. And in this case, the hatchery operators can easily use this avenue to at least rear them for few months or something to take the benefit of this technology. And even for the nursery rearing, it's a wonderful technology for nursery rearing. We propose it, we have to work out and check whether, I mean, it can be a wonderful technology for nursery rearing too. These are some of the publications we had during this period from our research from 2013 onwards. Few more are there. Thank you for your patient listening, and we hope we will go ahead in Indian aquaculture scenario using this technology. Thank you very much.
Well, thank you, Dr. Babita Rani, for sharing your experiences with the biofloc technology. Before we move on to the next presentation by Mr. Eric Ogello from Kenya, I receive a lot of questions, queries in the chat box. Definitely, Professor Yoram has agreed to collaborate with Fisheries Research and Information Center to do research on different fish species, Indian fish species, their stocking density and floc management and new strains, new bacterial strains which can enhance better floc in the aquaculture system. Friends, under the Pradhan Mantri Matsya Sampada Yojana, throughout the country, many entrepreneurs are looking for funding support from the government agencies to establish biofloc technology. Unfortunately, most of them do not have the hands-on experience. Soon after the COVID issue, at FRIC, Fisheries Research and Information Center, we'll be organizing hands-on experience with the support of experts from different countries. India, at present, producing 12.8 million tons of fish, and per capita availability is about 11.3 kilos. By 2025, through these support services from the government of India and different states, we are aiming to achieve the 15 million tons of fish production annually. Definitely, looking at the land resources, we have 150 million hectares of agriculture arable land, and we cannot afford to utilize these productive lands for fish culture because staple food production is the main objective. At the cost of staple food, we cannot produce more fish. In that context, biofloc technology offers new opportunity to produce more fish in a smaller unit area with limited resources. Though it is capital intensive, looking at the benefit cost ratio, it is worth. And but if you don't practice the right practices like management of oxygen, management of CN ratio, because carbon and nitrogen ratio should be more than 14. You can go up to 20. Believe me, 60% of the food in the aquatic system is less than 60 micron. Even fishes cannot harvest these smaller organisms because it will pass through the gills. If you maintain the right carbon and nitrogen ratio, they will flocculate, and that will become a part of the feed, and which will have more than 35% of protein, crude protein in the composition. So in that context, we can reduce the cost on feed by 30 to 40%. That is the main funda of the biofloc technology. We'll come back to you. You can have more queries and reserve your questions and send the questions in chat box. We are collecting all the questions and experts will be answering it. Now over to Dr. Suprita to invite Mr. Eric.
Yes, thank you, sir. That was a wonderful and an informative presentation, madam. Thank you so much for your time and information. We have one more presentation from Dr. Eric, aquaculture consultant, Kenya. It's my privilege to have a brief, to give an introduction about Dr. Eric Ogello. He's a scholar and research scientist currently working as a lecturer in the Department of Fisheries and Natural Resources, Maseno University, Kenya. Dr. Ogello has a Doctor of Philosophy degree in Fishery Science from Nagasaki University, Japan. His research interests include fisheries science, aquaculture production, biofloc technology for larvae culture and grower fishes, aquatic ecology and ecotoxicology. Dr. Eric currently running Kenya Climate Smart Aquaculture Project, Climate Smart Fish Culture Systems, and has published over 52 scholarly articles in peer-reviewed international journals and participated in several local and international conferences. Sir, kindly do present, sir. Dr. Eric, please.
Thank you for your introduction. I'm happy to join this webinar. I feel honored to be invited to this webinar. I'm just trying to share my presentation. I'm also very excited to see the father of biofloc technology, Professor Yoram Ablimelech from Israel. I'm very happy also to mention that we have been in close contact with Aquadoctor Solutions from India through NEPTA. And I'm happy that we have these connections to even share our experiences all over the world. Yes, as been mentioned, I have a lot of interest in biofloc technology. And now I want to present to you what we are doing in Kenya in regards to the biofloc technology. Please, can you enable me to share my screen? Dr. Magada?
Yes, you can. Meantime, I request Professor Yoram to see the chat box. There's a question about bioflocs. Red fish may have off flavor and more microbial load than normally cultured fish. We will answer it during the question or session. But kindly note all the speakers, scientists, a very pertinent question. Let's answer it.
Please, Dr. Magada, enable me to share my screen. I cannot be able to share.
Yes, you're enabled. You're enabled. Should we have to upload the presentation for you?
It's showing me that the host has disabled me to share the screen. So maybe you can share the screen from
Please
your
hold
end.
on. Please hold on for a few seconds.
Okay.
Eric, illige presentation. Host, huh? Now you are the host. Please, you can upload the presentation, please. Is it all right?
Okay.
Yes, it's coming. Fine. We are seeing your presentation on the screen. Please go ahead.
Okay. Okay, thank you. Thank you so much, Dr. Magada. Yes, welcome again. I'm happy to be in this webinar. My name is Eric, as been mentioned. I want to talk to you about fish farming with biofloc technology, the Kenya's perspective, how we are trying to understand and how to implement the biofloc technology to improve our fish production.
Please put
in
the,
country.
Dr. Herrick, please put
Yes.
the slides on a slideshow mode.
Okay.
Okay, now
Is it
all
clear
right.
now?
Please go ahead.
Okay, so I was mentioning that I want to speak with the audience about our experience in regards to the biofloc technology in Kenya to improve our fish farming sector. And therefore, I also need to mention that biofloc technology is relatively new in Kenya, and we are trying to adopt the technology with the youth groups and women groups, and also in government agencies, so that we are able to improve our production in fishery sector. I'm going to talk about briefly aquaculture in Kenya, and I will also present the biofloc technology in terms of our perspective. I will discuss deeply about the biofloc technology for live food production and laviculture. That's what I like most. I like to talk about that one very mostly. And then I will also share some experience from Aquadoctor Solutions, my friend from India, Naptam. I'm happy you are here. How we are trying to adopt a tank-based biofloc technology. And finally, the pond-based biofloc technology. I also need to mention that we are partnering with Textile Africa Industries in Nairobi. This is an Indian-based company, and our university is partnering with this company so that we are able to implement some of the biofloc technology. We, as you see later, we have not yet started on the pond-based biofloc technology, but we are already making progress in that regard. So quickly, just to mention, we all know that aquaculture production now globally has exceeded the capture fisheries. And this is attributed to the technologies that are already being experienced, that are already being employed in aquaculture. And we know that the population is ballooning and the fish consumption demand is increasing every day. And therefore, there's a lot of pressure in aquaculture industry to be able to supply sufficient proteins for the growing population all over the world. And the FAO has mentioned that aquaculture is increasing in terms of production for food consumption. And therefore, all the technologies that we are talking about, including the biofloc technology, are all geared towards increasing fish production for human consumption. Yes, in Kenya, we are currently producing about 15,000 metric tons of fish. Out of that, tilapia, the Nile tilapia, aquatic chicken, the darling of the press of the many people, we are producing about 85% of tilapia, and catfish is around 12%. We also have trout, which is probably less than 3%. We are producing this fish in ponds, in RAS, in raceway, in cages, and you can see biofloc technology is in red. That means that we are just starting, or it's still not very, very, very popular. As you can see, between 2011 to 2014, the fish production in Kenya was on an exponential growth. We produced about 25,000 tons of fish in 2014. And during this period, the government enrolled an elaborate project called the Fish Farming Project under the Economic Stimulus Project, where ponds were constructed for farmers all over the country. And there was a lot of fish production. What we are seeing from 2015 onwards is a decline. And this means that most people dropped culturing fish after the project ended somewhere in 2013 or 2014 there. and a slight increase in 2018. Yes, some people are trying to come up, but as you can see, we are not yet where the Asian tigers are in terms of fish production, and we will really wish to be able to produce the millions of tons being produced in India, as Dr. Magada mentioned earlier on. Yes, and therefore, that's why we are embracing the concepts of biofloc technology to help us to be able to produce fish for our people. I cannot mention it better than the father of biotechnology himself, Professor Abdi Melek, has really given a very passionate presentation there on the advantages of the biofloc technology. So I will not want to go into details, but I will want to highlight that this biofloc technology saves feed by about 30%. We have been told it saves water, it is biosecure, we have faster growth rates of fish, we have a shorter culture cycles, and the technology maintains good water quality. All these are indeed very interesting attributes of a culture system that is geared towards maximizing production of fish. Yes, and what are we doing currently? We started with live food production, and this is what I'm going to speak about in detail, because in Kenya right now, we have the biggest problem in terms of the fish seed production. And the disparity in fish seed production is more pronounced when it comes to the larval fish feed. As you can see in my slide there, the fish larvae normally has a very tiny mouth gap. And the stomach of the fish, the digestive system of the larval fish is very rudimentary, not well developed. In most hatcheries here, we provide a nut diet for the larvae, which the larvae cannot digest. And in some cases, it cannot ingest either. And therefore, most fish end up dying, and fish farmers and hatchery managers report huge fish mortalities in their hatcheries. So we are trying to adopt the biofloc technology and apply it in live food production so that we are able to produce enough live food resources such as rotifers, copepods, artemia, and cladocerans, so that we are able to have fish larvae with good survival and good health. And I will explain the next slide how we are doing this. Of course, we know a conventional way of providing or culturing the live food resources is normally through the algal pastes. But these algal pastes are very expensive and very, very, very difficult to culture because they crash a lot. So we have been able to provide a system of biofloc technology that we apply to produce live food resources. And I will explain in the next slide. This is how I've summarized it. We are using environmental wastes, mainly fish wastes. As you know, a lot of fish goes to waste and they end up affecting the environment. So these are the materials that we are converting through, I call it fish waste diet protocols. And we add some molasses, we do aeration, and sometimes we add natural bacteria, maybe yogurt or spoiled milk, and we inoculate some live food materials there. And also we use chicken manure extract, what I call CME. I will explain in the next slide how it works. Now, through this protocol, we are able to produce 80% bacterial flora, and also it produces about 15% of fine particulate matter. And of course, there are coarse particles forming around 5%. This is centrifuge bacterial samples. And these materials are providing food for the live food materials. These are the side you can see we have copepods, cladocerans, and rotifers. And we feed them with these materials, and therefore we are able to feed our fish larvae with the live food resources. As you all know, if we don't dispose the environmental waste, they end up as environmental pollutants. So in this system, we are able to solve environmental pollution, and we are also able to convert these environmental pollutants, organic pollutants, into usable materials that can be used to enhance aquaculture. So yes, this is how we do it. We, in about 500-liter tanks, asbestos tanks, or sometimes just glass tanks or concrete tanks, we provide the molasses here, and the fish waste diet. That's just the fish waste that we put it in some sort of net, and then we drop it inside the tank. As you can see, there's a string here that is holding fish waste down there. That is to provide a substrate for bacterial growth. And also we provide molasses here, and also sometimes we also add a chicken manure extract. And a little bit of aeration we do. And of course we inoculate with rotifers, copepores, cladocerans, and sometimes of course mosquito larvae will definitely appear there. So we call it actually live food dispenser, because after this, I will show in the next slide what kind of numbers we are finding in this system. We are able to provide aeration to the tanks, and all this is under greenhouse to stabilize the temperatures. And therefore, sometimes if we want to do special or let me say single species culture of the live food, we also are able to isolate a specific species, for example, rotifer only, and then we culture them using the biofloc technology in tanks where we can control environmental temperatures, and also we do aeration to provide oxygen, and also this is under a house or a greenhouse or under laboratory. So this is just a demonstrational unit on how we are able to grow pure rotifer cultures using the BFT technology. You can see here, down there is a black substance. There is where we are holding the fish waste, and the foam-like things are actually bioflocs or some forms. So we are able to culture the live food here, rotifers here, in very, very huge numbers. Of course, sometimes we use 30-liter tank. Inoculation, we are providing 20 rotifers per milliliter at the beginning. The temperatures we are maintaining at 30 degrees maximum, and these cultures normally after 20 days, we start afresh because they will crash. And of course, we check the oxygen level, and also we also check the pH level, the pH levels. Yes, we are able to find up to 1,200 rotifers per milliliter in these cultures. And as you can see in this graph here, after the first 5 days, we do harvesting. You see the black arrows, that's harvesting. So we harvest the water from the tank, and then we sieve that water. We only isolate the live food, the rotifers, and then we use them to feed our culture tanks where the fish are. And you can see the population goes down after every harvesting, and without doing any other thing, we only add more water. The cultures are able to grow again by themselves. And once we monitor the peak where they just start to produce, we do harvesting again, and then the system picks up again by itself. At this time, we are not adding any molasses, or we are not adding any rotifers. We, of course, sometimes we do add molasses if we feel that the population is not growing that fast. But what is interesting is 14 days later, we are normally seeing that the culture does not pick up very well. That means the system is tired, exhausted, and probably crashing. So at this time, we wash everything, and then we start our cultures afresh. But what is interesting is that sometimes we're able to find up to 1,200 rotifers per milliliter within 14 days, or after 10 days, or maybe 9 days, we can get around 1,000 rotifers per milliliter. That is relatively sufficient to run hatcheries that are not producing a lot of fish in rural areas. And we are also starting to embrace tank-based biofloc technology, where we are using we are using these ones to grow fish, grow out fish. And I must say we have been partnering very closely with Aquadoctor Solutions, my friend Dabtam from India. Yes, and therefore we have actually adopted his protocol for bacterial preparation. We do make a concoction to make bacteria. And you can see here, this is a group, a self-help group in one of our counties in Kakamega, where they are trying to use biofloc technology to enhance their fish production in ponds. So they have this tank here, and they use some manure or some yogurt, they add there, and they do it in a very traditional way. So maybe they don't know much about what they are doing, but basically this is biofloc technology. So we visited them, and we have recruited this community to work with them in our project that we have just started to roll out. So yes, this is borrowed from Aquadoctor Solutions. We, of course, advise that 250 to 300 grams of molasses is sufficient to maintain a CN ratio of 10 and above, as other previous presenters have mentioned. So this bacteria is grown in a 20-liter culture tank before it is added to the main pond. So we have a project currently. We are validating biofloc technology as a climate smart fish cultures technology in Kenya. And I'm the one leading this project in our university. So we are working with youth groups, women groups, and some members of the society to be able to actualize this technology to improve the fish production. Some pictures from Aquadoctor Solutions, how to construct biofloc ponds. And yes, we are just about to start this one. And this is now showing how a completed pond is working, sometimes in open or sometimes in closed enclosures. And we know that in open air, we get green flocs, but in closed systems, we get the brown flocs. Of course, the pH has to be maintained as at 7.5 to 8. And also the total dissolved solids must be checked, and the range is provided there. The tanks, the size of the tanks can vary, but mostly we prefer 10,000 liter tank onwards, and then the bacteria is inoculated. So we have learned a lot from Aquadoctor Solutions in terms of making tank-based ponds. Some of our farmers have already started making the ponds, but not yet production at the moment. I also have students which are working directly with me in my laboratory so that they can be able to actualize this biofloc technology. They understand what it is. And one of my master's students is doing biofloc system as a way of improving protein utilization in Nile tilapia in one of our research centers. So you can see the student is, you can see the student is trying to use the glass aquarium tanks and explaining to other students how he's able to even use different types of carbon sources. So the student is using molasses and also using glucose and rice bran. So trying to see which carbon source is best in terms of maintaining high carbon nitrogen ratio. Checking the water quality parameters and also monitoring the bioflocs themselves and other aquatic lives that are developing in the tanks. Also, we are partnering with the textile industries Kenya. This is an industry based, a company based in Nairobi, and they have specialized in biofloc, in pond biofloc technology. So we are partnering together. Soon we are going to establish this kind of ponds you are seeing on your screen in this project, so that we are able to produce fish for consumption, table size fish for the masses. And we are proposing a 2,500 meter square pond, which will be fitted with the necessary technologies and equipment to be able to produce fish in this biofloc technology. I think this is going to be the first time that perhaps Kenya will be able to demonstrate how pond-based biofloc technology can be able to be used to enhance fish production. Of course, some managerial skills in biofloc systems that we have learned. Floating feeds is, of course, good, as is also in other systems. Starting with 30% crude protein, which is probably 6% at 6% body weight for the first 3 months. And it goes on. Also, we monitor the biofloc every week. And as mentioned by the father of biofloc technology, that we need to be very cautious in terms of managing the biofloc systems. That's very, very important to look at oxygen and to maintain optimal sludge. That is sludge management. Very, very, very key. I was very happy to hear that from Professor Yoram. So we are looking forward to, minimize the harvesting cycles and be able to produce fish within the shortest time possible. Thank you so much. My presentation will end there, and I want to take it back to Dr. Magada to be able to proceed. Thank you so much.
Thank you, Dr. Eric Ogello. Definitely, we can do a lot of works. And as Dr. Ogello mentioned, the most critical stage in the fish life cycle is the weaning stage. During the early stage, if you don't provide the right food, which fits into the gape of the fish mouth, they will die in large number. This is called a Zod critical period. During this weaning stage of a larvae, most of them will die. And sometimes fish food organisms will be bigger than the fish larvae. So in that context, if we can flocculate the microbial flora, plankton flora using CN ratio in a right proportion, they will be able to nibble, and digestibility of this biofloc is much better than the conventional diet which we provide. Normally in Indian context, we provide groundnut cake and the rice bran. In fact, during the first 10 days of hatching, if you feed the rice bran, it is complex cellulose. Fishes cannot digest because they will not be having the enzymatic digestive system. It is still rudimentary. In that context, biofloc will help us a lot. And let's hear from our scientist from Bangladesh. Over to Dr. Suprita. Please introduce the scientist from Bangladesh.
Yes, sir. Thank you, sir. That was an excellent presentation, sir. Thank you very much. Now we have presentation from Zillar Rahman, aquaculture consultant and founder, Genetic Aqua Bangladesh. He's a former tilapia breeding program specialist, World Fish Bangladesh. Currently, he's serving as a managing director, Bluefin Foods and Services Private Limited, and heading one of the largest project of biofloc. I now kindly request Dr. Zillar Rahman to present. Thank you.
You can hear, everybody can hear?
Yes.
Thank you very much, especially Shiv Kumar Magata. And also I'm very much happy and grateful to invite me for such a program where the father of biofloc delivered his speech. We already got 4 presentation. Yoram, Bobita Rani, Dr. Eric. In our country, especially Bangladesh, we are this is the initial stage for implementation of biofloc technology. From 2019, this is just beginning, and in 2020 we start some large-scale biofloc also, but still it is just beginning, not up to the mark. And I think as we have many pond farming, inland water fish farming, so still biofloc is not popular up to the mark. And also the price of natural fish and also is very less. So somehow we are trying to establish the biofloc technology in Bangladesh. I am very much happy and grateful to Dr. Dev Tono, Dr. Solution. He also tried and visited many times in Bangladesh to introduce this new technology, and we some other guys already started to establish the biofloc technology with the basic principle of Yoram technology, Yoram guidelines, this principle. So this is just beginning, but in Bangladesh, tilapia and pangasius fish is, price is below $1. And in the production cost of biofloc farming still not up to the profitable mark, but somehow we are managing to establish the technical viability of the biofloc system. We already established tilapia farming, climbing parts, Vietnamese koi farming, and also some other catfishes like shing, papda, gulcha, tangra, like that. But this is, I think, not up to the mark. From my observation, in Bangladesh, in aspect of Bangladesh, if we continue the approach, integration approach, like adjacent nursery, biofloc hatchery, there are so many systems for same species, many tanks like broad tank, fattening tank, and link nursery, and also good quality fry fingerling from the hatcheries. So technically now we are we can produce a maximum species in biofloc in Bangladesh. But as I mentioned, the price of fish, in Bangladesh, tilapia only $1, koi $1.2 like that, or more than $1. Catfishes like then $2 and $2.50. But India price is very high. So India, rather than Bangladesh, India rapidly growing the biofloc system. Some university also they are trying to establish the biofloc system. But in our pond farming or inland aquaculture system, still production cost is less than biofloc system. So we need to establish the branding of the organic fish, live fish, marketing system, integrated approach, like then we can reduce the cost of production cost of the biofloc farming. Then ultimately, and in urban aquaculture, we have huge population. In urban aquaculture, we have great potentiality in Bangladesh. We know we are fourth like that in inland fish production, fourth in worldwide. So huge consumption, huge demand of fish. But if we can establish the organic branding, live fish transportation or fish processing and also export, we are surplus in production of fish in Bangladesh, like 44 lakh metric ton in now production. But our requirement is 43 lakh metric ton like that. So we are trying to establish the biofloc system. I think there is a great potentiality. The farmer who are very much skilled, who are running normal aquaculture like intensive pond farming, intensive nursing system, they are now capable to manage. But the farmer who are very much interested by seeing the YouTube or like that, they are just involving biofloc system. They even do not know the uh, fish of which is fish in urban young generation. So I suggest, we suggest that you know first the fish farming system, then know actual biofloc system, water quality, aeration system, everything, then integration approach of link nursing, fattening, grow tank for the same species, and starts market system, find the fish value is available, fish market price is very high. So everything all together, I think if we can work and exchange our views time to time, like this type of webinar, at least we can know more about the biofloc culture system. If it is technically viable, I think tomorrow we can find viable by financial viable, but need some time. So I am very much happy and grateful to the organizer and also the other participants that I can attend the webinar and deliver my little space. I'm very much grateful to the organizer. So thank you. This is the present situation of biofloc system in Bangladesh. But few urban farmer in young generation, they are trying to establish biofloc system. And now we are arranging different organization awareness campaign, workshop, and exchange visit that you learn first what is the biofloc system, how can reduce the cost, how can integrate the approach, and then you go for commercial biofloc system. So this is the practical situation in Bangladesh. Thank you again for giving the floor to say something in this big floor. And it is my pleasure then I can attend the program with the father of biofloc. So thank you very much everybody again, and we are grateful from Bangladesh, and we are very much happy our many farmers attenders registered for the program. I think they will be also helpful. They will be also benefited from this program, and in future we are hoping we can attend such type of program, innovative program in future. Thank you very much. Thank you very much.
Yes. Thank you very much, sir.
This is from my side. Thank you very much. We can go for further.
Yes, sir. Yes, sir.
Yeah,
Thank you very much, Jilur Rahmanji. Next is Mr. Ramesh. He is aquaculture consultant. He is trained in Israel and working at Nigeria and doing biofloc farming with tilapia and catfishes. Mr. Ramesh, are you there?
Yeah, I'm here.
Yeah, please
Can you hear me?
share your experience. Yes, yes.
Okay. Thank you very much.
A small announcement before Ramesh speaks. While introducing Mr. Jilur Rahmanji from Bangladesh, it was wrongly quoted with the designation and expertise of Mr. Hitendra from Karnataka. Sorry for the inconvenience. Mr. Ramesh, please continue.
Good afternoon, everybody. Thank you for giving me an opportunity to speak some of the experiences which I have. First, actually, most of the speakers they covered all the basic things which are required for the biofloc technology. So I will not go into detail about that. So I would like to speak something about the biofloc technology in Indian context and how we can do it. The biofloc technology which has been introduced is a very good technology, but we have to decide first whether are we using it for the uh, only for clearing the water or maintaining the water quality or to feed the fish. Then we can decide about the species. For example, if you are going only for the water quality, you can even do carnivorous fish with the biofloc technology. In case if you are going with you want to do the both the things, like you have to feed the fish at the same time to maintain the water quality. You have to go with. Can you hear me? Is there any problem? Yeah, okay. So then you can go with the species which can feed on the biofloc and also it can help in maintaining the water quality. So it is always beneficial to go with the species which can feed on the biofloc and at the same time to maintain the water quality. And at that time, you need to consider whether biofloc technology will add any benefit to us. Like, is there any, ultimately the benefit means what is the financial implications, whether it will be beneficial or it will be adding up to the cost. That we need to first find out. And as far as my understanding of Indian context goes, so far we can go ahead with the shrimp farming, that is the Vanami culture, we can go with the biofloc technology. And other candidate species, what we have is tilapia, that is gift tilapia. For shrimp farming, to certain extent, we can go initially because when we are doing with very large scale, up to let us say in few million tons of tons, in growth stage, it is not at all possible to maintain the good biofloc culture. In early stage, like in nursery stage, since we are doing it in 2-3 stages, like in nursery or juvenile stage and all those things, in juvenile stage, we can go for the biofloc technology. Later, we need to shift it for the normal culture system. And in tilapia also, as far as now, we do not have good market for tilapia, but definitely when culture progresses, we'll be having other ancillary industry which is coming up, like we'll be having the processing plant exclusive for tilapia, marketing facilities, like we can market in fillet form instead of selling fish and selling raw fish, or we can go for live fish sales and all. With that, we can go ahead with tilapia in Indian context. And some of the basic things what we need to understand is, first we need to see the water availability of the water. If you have sufficient water, then you can go for with biofloc, you can go for a certain extent, then you can shift for the normal water. Or then the other thing is availability of power, electricity. Now, as of now, the important factor which holds constraint for the biotechnology is the power cost. Normally, earlier days, we used to talk about the cost of production of the fish. Like 70% of cost goes for the feed and all we used to discuss. But now, the feed cost has come in biofloc technology. Feed cost will be 50% of the cost. Then, but the adding the carbon, that is carbon source, it will cost you around 10%. Then again, power, it will cost around 20%. Then remaining labor, the other things, it will cost another 20%. So that is the consideration we have to take into account while going for biofloc technology. Power is the most important factor. Then they having the aerators or whatever type of aerators, always in Indian, what I found is blower diffuser system of aeration is the best for the biofloc rather than having the paddle wheel aerators. Paddle wheel aerators just it will do the circulation. It will not keep. One thing we need to notice, like we need to have a floc in suspension in the water for always it should be in suspension. Once floc gets settled down, that is end of the culture. So to have the floc in suspension, we need to have sufficient aeration. That is with diffuser type of aeration. That is the second important thing what we need to, and it should be always 24 hours. That is 24 hours aeration is required. That means you need to have power backup for 24 hours. If you do not have power for at least for 15-20 minutes also, you may lose your stock. Then earthen ponds, we cannot do the biofloc. We need to either should have the concrete tanks or with the plastic lining, or some of the tanks like what we have, the circular tanks, which can be easily installed or erected. Those type of tanks are better. And this the normal size should be for commercial farming. The size should be at least, it should hold 30 cubic meter per water. 30 cubic meter water, minimum size should be 30 cubic meter water. Very small tanks, it will not be for as a theoretically we can do it, but practically when we go commercial, when we do, at least 30 cubic meter water is ideal size. Then when we are going for the biofloc technology, it is always to have it in stages. Stages means you should have 3 different sets of tanks. One is for growing your fingerlings, that is your nursery. Then you have to shift it for juvenile size. Then the final stage is growth. So that will help you to. This will be having its own advantages. Like you will be able to segregate the fish well. You will be able to know the number of the fish. That will be helping you to adjust the feeding, and also you can apply the biofloc technology where you need. For example, if you are not going ahead with the biofloc technology growth, you can stop there. You can do it in nursery and with stage and all. So it is always better to have 3 stages in culture. So in a nutshell, I would like to close, because I do not want to run my slide, because after the slides have been covered by the experts. So in a nutshell, as a technology, biofloc technology is very good, but we need to specify, or specifically we have to do it for the areas like in Karnataka what we can do, or in Northeast what we can do, or in Punjab or wherever what we can do. It is very site specific, because the source of the carbon, whatever we are adding to the system, it will cost you around 10% of your production cost. So if I say molasses only has to be used, that may be in sugar growing areas, it may be very cheap. In other places, it may be costly. So we need to look into different carbon sources which is available cheaply in that area. And instead of adding directly the sugar sources into the water, we need to ferment it. That means we have to break down the carbon so that it will be easily available for for the assimilation in the system. That is simple technology. What we need to do is fermentation, 2-3 days fermentation. That will by adding some bacillus or your yeast, will be able to ferment the sugar source, and whatever supernatant, whatever is available, that can be added, and whatever the bottom the sediment, that has to be discarded. And the other things already we discussed. The sludge. Sludge, it has to be removed. The most important thing is removal of the sludge in the sludge in the aquaculture system. So either we can drain the sludge by using a central drain, or we can even use some sludge pumps to just siphon the sludge out. So how to know the sludge, whether it is there in the bottom or not? Normally there will be quantum of sludge which will be deposited in the bottom, but to remove to know practically when we can just go inside the tank. If it is bottom is slippery, then definitely there is a sludge that has to be removed. Then how to add the carbon. Carbon we can add 2 ways: by calculating. There are tables which are available already in many of the YouTube channels and all. Either by calculating the carbon by calculating the percentage of protein in the feed and quantum of the feed which is going to the system and all, or we can measure the total ammonia which is present in the aquaculture system by using the digital meters, or by using the titration method and all. So whatever the quantity of ammonia which is present, we can add sugar by multiplying it to depending upon the ratio what we need, carbon nitrogen ratio 10:1, 12:1, 15:1, 20:1. And the initial stages, it is always better to have maintain the floc density of around 5 to 7 ml per liter. It can go up to 10 to 12, but if it is more than 12, then it is very difficult to maintain the system. It is very I suggest the floc density should be varying from 5 to 12 per ml. That is better to manage, and it will be we can maintain it constantly. Then other than this, I think if there is any any questions or queries, you can directly contact me at Dr. Shubham Magara, the hosts who have done this. And by this, I would like to conclude. I do not find any other things to talk now. Thank you for giving me an opportunity. And please, I'll hand over the proceeding to the host. Thanks.
Thank you, Mr. Ramesh, for sharing your experiences. Definitely, you have raised a very pertinent issues like cost of production, cost of electricity, and cost of molasses. In fact, in many of the states, molasses is not available freely because people might misuse it for the production of country, sarai, and all. And the cost of molasses also, because most of the sugar factories who are producing molasses, they'll be using it for their own alcohol production. So in that context, even molasses will become a critical input. And electricity, you said it, the cost of electricity in Karnataka, it's about 7 to 8 rupees per kilowatt. By considering all that, if you don't understand the market dynamics, and definitely biofloc system will increase the production. If you don't understand the purchasing power and consumer preference, definitely you cannot place your product, because when we worked out very recently with the biofloc system, the cost of production will be 107 rupees per kilo. If you don't know how to place your product, saying that it is safely grown, hygienically grown, if you don't know how to price the product, compare with the native varieties which are available for lower prices, you will go under loss. So definitely you have to have your own channels to sell the fishes. 30% of your produce, you should be able to sell it live for 250 rupees and above. Then only the average cost will be around 150 to 160 rupees. Otherwise, by producing more fish, you cannot look for the market. So in this context, I request the to form a farmers producers organization, or to have a network with the Metro, Spar, such other multinational companies who are selling the fishes in a hygienic manner, and supply them consistently and get the better price. We will come back to that economics. And now I request one of the successful farmers, Rajiv ji, are you there? Rajiv Kumar Deer from
Thank you.
Bihar. You will going to speak in Hindi, and somebody can translate it. Jitendra, you can translate it against the demand. Rajiv साहब, आप ऑनलाइन में है? Rajiv, Rajiv, check this. Rajiv. Okay, go on to the next, please.
Okay, thank you so much, sir. I now request Mr. Hitendra PV, Biofloc Consultant, to kindly present. Mr. Hitendra is a Managing Director, Bluefin Foods and Services Private Limited, and he is an academic trainer, former and researcher, expert on water quality management, and currently he is heading one of the largest projects of Biofloc. Mr. Hitendra, please.
Yeah. Hello. Can you hear me?
Yes.
Yes. I would like to share my screen.
Please.
Could you please give me access?
Now you are allowed. Please share your screen, Mr. Hitendra, please.
Yes, sir. Sure.
Mr. Chandra,
Yeah, yes, sir. I'm
please
just
don't feel
just a
bad.
minute.
When we were reading the testimonial of the speakers, it was printed on the same sheet. So that's what it
Yes.
was wrongly mentioned with the Jillur. I'm
No,
sorry.
no, that's okay.
Okay,
That's
okay.
okay. That's absolutely okay, sir. Can you please can you see the screen?
Yes, yes, quite clear.
Yes. Yes. So I personally am into aquaculture. I've been into aquaculture since my uh, right after my college, in fact, agriculture and aquaculture. But as a hobbyist, I was there from my childhood, right? Into aquarium keeping and all this. So this technology of biofloc, we have been working on this since almost 3 years, 3.5 years, I would say. And I've been it's an excellent opportunity, and it's a big honor for me to share stage with such bright minds and stalwarts of this field, Dr. Yoram and so many others. I'm really happy for that. So many of the science behind biofloc and the nitty-gritties of management have been already shared with the other participants have already spoken about it. I would like to present the work that we have done. So this particular work has been done in a place called Koppal. It's a district in Karnataka, Koppal district. So here we have been this project is about a year old right now. So we have been able to take the first harvest, and right now we have been selling the fish. So the different aspects that I have come across practically and faced challenges and have come across, I've overcome it is something to do with first of all, we had to work on the tank geometries, so the geometry is very, very important because again, as Mr. Ramesh was mentioning, the power costs and the power costs in Karnataka are especially high. So and also if you look at the terrain that we have constructed our ponds, so here conventional aquaculture cannot be done. You cannot do pond based aquaculture. We have to go with a liner because it's a rocky and stony land, and even vegetation is sparse. It's not a very good area for crop culture. So here we had to choose a technology which is which was based on water conservation also. So that's the reason my partner and me, our team, we decided to go ahead with biofloc. So here, the first thing that I would like to touch upon is the geometry of the tanks. You see, if you look at the layout of our tanks, right, we have a nursery system up front. There's an upfront nursery system, and then behind the nursery system, we have all our growth ponds. So the nursery system, the tanks are of different dimensions, which will help us, if we get seeds which are 0.02 grams and all. Especially, we are working on tilapia. As of now, we have been working on tilapia. So when the seeds are very, very small, right, you'll have to take it through different sizes of tanks to get them used to the feeding cycle and also management. There might be fishes, there might be mortality and all this. So as on a daily basis, operations become very tough if you directly put them into bigger tanks. So we have actually designed this system based on our production requirements. So we work back. So this plant is capable of producing about 250 tons a year. That is our production target. We haven't still reached it because it's as I told you, it's just a year old, and the cultivation period has been about 7 months. So right from the groundwork, it's been about a year now with this month. So the seeds, we have to take it through different tanks to keep the management easy and the labor costs low. So that's how we have designed the system. So this upfront, what you can see is the nursery system. So we have tanks of different dimensions and different cubic meter capacities. So what happens here is, so if it is above ground and if it is a circular tank, one thing is if you generate a current and a water movement, that can be easily maintained in a circular tank. And the nursery system being above the ground helps us in keeping away other predators especially predators, which can can be easily managed. And see, the cost, because the circulation is easy and neat, the cost of electricity, right, the amount of horsepower that we spend on each tank is reduced. Okay, so the second aspect why we use circular tanks is I want to also talk about aeration system. Probably I would just like you to just look at the land which was earlier there and the transformation which it has gone through with our work. So this was the land, just the empty site when we came here and started the work, and we took it through its different paces. So our company, me and my partner, we both have worked on all these aspects of getting the tanks. I mean the buying the material and then getting the lining work done. The different style of welding. These are HDP membranes which we have used. So different style of welding these round tanks, and also doing the earthwork, the concrete and the whatever the masonry work. So the everything from scratch we had to do, and we had to train people who are working on this project with whatever we could sort of at that time get our hands on. So these are the way the grow tanks were prepared. As I was mentioning, this soil actually is very loose and gravelly here. So in order to compact the tanks, we had to get in heavy machinery also to do it. So these grow tanks which you see here, it's the capacities are I mean it is about 2000 cubic meter capacity, that the volume will in terms of liter will sort of go to about 20 lakh liters, 15 to 20 lakh liter capacities. So the plan was to do biofloc in these huge tanks, because as I have also come across small tanks, right? Initially, when we started off with small tanks, they were not making economic sense, because of the high cost of power and the quantity of fish that we could generate. And if you have the cost per unit would be very large, and to recover that cost per unit, you had to go through a lot of cycles of production, which really doesn't make sense for a large commercial operation. So this is about tank, which is about as I told you, it's about 2000 cubic meter water volume capacity. So it's about 1200 square meters of land area, and we have kept it circular. The reason being to reduce power costs. So the per HP needed. So there is a calculation based on biomass. If you have so much biomass in your tank, this would be the aeration requirement. So we would calculate in terms of HP. And again, your different aeration systems also makes a big difference. So here we have used pedal wheel, we have used venturi, and we have used diffusers using root blowers. So we have used all the systems. So at different densities, right? We activate a different aeration systems to keep the floc in suspension. The idea is to continuously oxidize the waste. So the oxidation process is helped by the bacterial enzymes, right? And this to keep this efficiency going, you need to use as minimum power as possible, because as Mr. Ramesh and Dr. Shivkumar sir also were mentioning, power costs are pretty high. So that's the reason, if you get the tanks to be geometrically correct, so the suspension of the floc and the aeration, all these you can have a good amount of savings. And and you see the floc are of two types. Earlier, I think this was one point that I would like to sort of focus on, because the other constraint which people have mentioned here is the use of molasses or any carbon source, right? So the carbon source being expensive or not readily available, okay, that creates a lot of problems. So you have to maintain a balance between your heterotropic and autotropic system. It need not be 100% heterotropic. If you go 100% heterotropic, then the system is completely going to depend on your carbon input. It's going to depend on your carbon input, wherein the availability of carbon and the availability of the raw material is one thing, and the cost is the other thing. So based on the density on which you go, the biomass on which you go, you can sort of every day tweak the system to maintain, say, 40% autotropic, 60% heterotropic, or 50-50%. So this is one thing that you can continuously tweak. And if you look, all our ponds are in open sunlight. So I don't think I have not come across any big problems with the pond being in the open sunlight. As you know, yes, that is true, that if it's on open sunlight, you have your algae growing, and your system tends to easily go towards autotropic system. It easily tries to migrate towards autotropic system. But here again, we can, based on the densities, right? The system itself, see, there is an equilibrium in the system itself, which will try to always go towards the well-being of the fish. Each of these organisms can have the potential to tweak it. We have to just interfere in such a way that the balance of the system is maintained. So whenever needed, we do add a carbon source. Okay, we do add carbon source and see that the water quality is maintained. Whatever the pollutants like your nitrites and your ammonia and all, to reduce these and to keep the concentration densities of the floc, we do add it. And every day there is monitoring happening using the Imhoff cone. We see by visual experience, we try and tweak the amount of algae and amount of bacteria. That is your to speak in simple terms, the green and the brown floc. So we try to maintain an equilibrium where we are not spending too much on the carbon source, and also there is no buildup of toxins in the water. And the other aspect is the electricity. So if you have your autotropic, or your photosynthetic algae which is present, even DOs can be maintained. Your morning and night DOs, you can tweak it. So this will take some experience. The handling something like this will take some experience, but someone who is working on it on a daily basis will eventually get there. So it is very important that the costs, as everyone's mentioned, the costs have to be regulated, because if it is a commercial operation, we should look at how much we are selling. So all the input costs have to be regulated, and whatever is absolutely needed must be done. Nothing more than that. So the other aspect what I wanted to also talk about was your, so this is one more slide wherein you can see these are all the grout tanks. So initially it was the earthen work was done, and then the liners were put, and the liners, there's a particular way you cut the liners to achieve this sort of geometry. So that has been done here. So that is one thing. And yes, the other thing what I wanted to talk about was sludge management. You see, sludge management is like a ritual in biofloc. So on a daily basis, there should be a dedicated person or a team who does this job in a commercial biofloc operation. Sludge has to be removed on a regular basis, and the quanta of sludge removed, So when we use aeration systems, so we are looking at the densities of sludge, So the heavier particles of sludge matter because, we control the flow rates of water. The water has to circulate at a particular speed. If this we maintain in such a way that the heavier particles which are going to be harmful for the system are flushed out. So in a day there is 1 or 2 times where the sludge is pumped out. So there is no pumping as such. This we also did because we wanted to get rid of the electricity cost. So in our farm, it happens through gravity. this what you have here is a sludge tank in front of you. So the sludge tank is at the lowest portion in the farm. So what happens here is, so whenever the valves are opened in either the grow tanks or the nursery tanks, all the water gets accumulated into this sludge tank. And in the sludge tank, there are a couple of species which further they can extract the juice, right? So everything is a spending, the feed, the flock, everything is costing us. So there are a couple of fish in this tank which further eat away the dead flock and all, and they can also sort of put on weight in a longer period of time. But the sludge, the majority of the sludge is pumped into the field. So on the very same farm, we have dairy as well as sheep farming happening, and because of that, we have a large amount of fodder which is being grown. So for this fodder, the sludge is being used, and so the cost effectiveness of the operation is again controlled at the last step also. So this is the main operation phase of the farm, and the other aspect is so you see, this to handle a biofloc farm, it is not very easy. I want people to understand that they randomly cannot jump into operations like this, especially commercial operations. If it is backyard farming, or if it is a small scale farming, you don't have a lot of you won't see a lot of losses happening within a small amount of time. You have you can bear with it if it is done more like a hobby. But if it's a commercial operation, a neglect of a few minutes also can cost you. So on this very farm, we have backups for everything. There's a backup generator. So even though we have a 24-hour electricity line, current isn't 24 hours. We have to use the generator. So in the rainy season, the diesel costs spike. So you have to consider all these all these varying costs, and over the period, you'll have to sort of you have to get into a spot where it is most conducive for your operation, not for someone else. Maybe someone else is selling the fish at a higher cost. Maybe you're still making, say, 20 or 30% profit. So in these situations, you will have to mitigate costs very in a very systematic way, and it takes in the beginning, it takes a lot of attention. So always you have to be there and keep control over all the aspects. So I just wanted to show my growth tank, if you don't mind, if you have a couple of seconds. As I'm staying on the farm itself, I can just walk towards the grow tank, and I can show you what exactly we are running in grow tanks, because that's where most of the nursery is something which is okay to handle. I just wanted to show how grow tank, the condition in which our grow tanks are. So no, if you I hope you don't mind, sir. Can I do that? Yeah. So the next aspect that I wanted to speak about is the daily data maintenance farm. You see, there should be a technician I'm talking about large commercial operations. There should be a technician who goes through, who collects the daily data that is presented in the farm and represents it, because you have to go through how much fish is there in your tanks, and what is the mortality rate, what is the feed rate, the FCRs. All these have to be calibrated on a daily basis on the farms, keeping a track of all this. And yeah, we go through the daily routine of sorting fish, and taking them through different stages. So this is our red tilapia. So this is what we have produced, and the marketable size that we have achieved. So harvesting becomes very easy with these, because if it is mud tanks and all this, the netting it and the netting and harvesting is a big problem. But in these tanks, harvesting is pretty easy. So there's a central drain in each of these big tanks. In every tank of ours, there's a central drain, which you can use to just flush the water out. Mantesh. Yeah. So there is a lab wherein all the parameters are regulated on a daily basis, and things are checked. I mean, all these many people have spoken about it. I don't want to elaborate. And the other thing that we are trying to work on is scampi. So we are using earlier we were into scampi production close to Bangalore, and that was in earthen ponds. Right now, I'm trying to use the same floc which is being produced in in biofloc uh, for for feeding the scampi and nursery unit also. I think. Previous speakers have also spoken about it. Yes, it is a very good source for especially small fish. So the scampi, we have been able to grow scampi using the flock, and if you can see, this is the sizes that we are sort of got. This is a female scampi. So we started off when it was very very small. We were able to sort of get this particular size. Yeah, and I just wanted to show you our growth tank. Could you please tell me if it is visible? Could someone please tell me if it's visible? I'm not able to see the screen. Hello. Yeah, this is our growth tank, wherein yeah, we are using mechanical aeration methods also. We are using a paddle wheel aerator with an injector, and also there are diffusers. So I just wanted to show you uh, show you the density. So we have maintained huge densities in this particular pond. This is almost the second phase of cultivation. Second phase of cultivation. So we have been able to achieve a flock in the growth tanks also. Uh, I mean, as I told you, right, the molasses and addition of molasses and electricity has to be carefully tweaked. If you see, this tank is both has autotropic and heterotropic system going parallelly, so that our costs are mitigated. And yeah, and tilapia being a pretty resistant fish, you can sort of stretch them a little. They are not very soft fish. Yeah, I would like to end here. I hope it's been useful, and I have been hugely benefited because of associating with Dr. Shiv Kumar and Dr. Manjapa and all these people who have sort of helped me throughout this journey. Yeah, thank you. Would like to end the presentation here.
Thank you, Mr. Tendra. We're very proud of you, and we'll work together because we need to understand many more dynamics in order to reduce the cost of production.
Yes.
And,
Yes,
dear,
exactly.
dear participants, as Mr. Hitendra said, we need to understand the engineering part of the pond, because by constructing a right shaped, right sized farm, we'll be able to accumulate the sludge using the aerators or the diffusers. We'll be able to collect the sludge and easily it can be pumped out. If you don't know how to, because people I saw in a chat box, people are talking about ferrocement concrete tanks, HDP tanks. For a biologist, we don't insist upon any of the structures. We need water holding structures of a right shape and right sized. And definitely biology will work on. More than the biology, farmers and entrepreneurs should be able to understand the economics part of it. And Hitendra and Mr. Ramesh, they said it very rightly. We will come back to you with some more details. Now, over to my colleague, Dr. Suprita, to introduce Deptano
Yes.
Parman,
Yes, sir.
experiences from Northeast region. Yes.
Thank you, sir. Thank you, Mr. Hitendra. Thanks for highlighting the important aspects of biofloc technology and possible troubleshoots and its corrective measures for the respective problems. Many thanks for sharing your practical knowledge. Now we have a talk by Mr. Deptanu Barman, aquaculture consultant, Northeast region, India. Mr. Deptanu Barman.
Yes. First of all, very good morning to everyone. It's my proud privilege to be here, and thanks to organizing committee, especially Dr. Shiv Kumar sir.
Little louder, Mr. Barman. Dr. Barman, yeah.
Uh, it's my proud privilege to be here, and I'm very happy to be here with all the experts from throughout the globe. And I'm very happy to Dr. Shiv Kumar sir to give me this platform to say some of my experience while working with different region in India, and also my neighboring countries, where I'm trying my best to be popularize this biofloc fish farming technology. And first of all, I like to put my sincere thanks to Professor Eram, because he is the father of this biofloc technology. And from reading his book, understanding the local issues in India and the Southeast Asia, I try to change some of the ideas into, backward socioeconomic aspects of our own people. I like to share some of my presentations in a slide presentation, so that I can show the audience like how I'm trying my best to popularize this unique technology, where we want to address food security along with the employment generation. Like the young people like me should come up to this industry to make our country self-sufficient in food sector. Sir, please, I can share it now, sir. Yeah. Understanding that today importance day, I put the title also, food security and employment generation through biofloc fish farming technology. I am basically a fisheries professional, did my studies in fishery science and work with many countries. And now I run a company named Aqua Doctor Solution in India. My company based at Kolkata in West Bengal, but I am basically from northeastern states of region in India. I'm basically from Tripura, but I'm now propagating this technology throughout the northeastern region through scientific interventions. So let me show you some of the things what we are talking. Many people talk about the science, how the tank looks, how the things looks, but I look for only one aspect, that is economy. Because any scientific approach and the intervention will be success unless until you know their economics. I will try to show some of the success story of some of the farmers, how much fees they put, how much price they got, so that people should understand this is a profitable economical venture by which we can earn our livelihood, and we can also give the job opportunity to many people. So in a simple things, if you just try to look, understand, in biofloc fish farming, we need to understand how much fish we can produce from a small tank. So I'm targeting here a 10,000 liter of tank. A minimum we can able to produce 300 kg of fish. And I try to put some numbers of the rate. Maybe this rate is not equal to every states of India, but I try to put this rate in on average for the northeastern states of the India. And total expenditures, profitable margin and all, everything I try to put here. I'll show the farmers what they are getting. And this is for one single crop. So I'm trying to calculate 6 months in one single crop. In a year, we can do 2 cropping cycle. Let me go one after another. So this is one successful farmer from Tripura. If you just see what he is getting, size of tank is 10,000 liter of tank capacity and tarpaulin based systems. And is a koi is Vietnam koi, and tilapia he stocked with IMC of fry size, and stocking density, you can see the numbers. If you see the numbers and if you go to the next level, you can see here what kind of feed he give, what feed percentage he have given, what is the cost of feed, because we understand that in northeastern states of India, the feed price is very high because we are a bit remote side of our India. So the cost of feed price though is very much, is 60 rupees per kg. But if you see the production price and how much we are getting out of the system is much more than what we are expecting. So this is a one success story of one farmer. I will show you 3 to 4 more success story of farmers, because details, whatever I try to put everything details so that anybody can read it out and can understand the systems. Next, Miss Sri Manik Majumdar. He's having one tank, and is a cemented tank. Is a 10 into 15 into 4 feet cemented tank. He started with Vietnam koi with 6,500 numbers, a rearing period of 3 months. Let's see what he's getting. So is it 1 mm to 2 mm feed, 2% of body weight. Cost of feed is 80 rupees per kg. I always mention the feed cost is very high in the northeastern states. And 120 kg he produced, though he got some of the issues of diseases, tail rot, fin rot in the very first culture cycle. But though the issue of sludge material management also, though he's able to get a good profit out of his system. Next is Sri Rakesh Majumdar. If you see, his cycle is a 2 tanks, 10,000 liter. Is a tarpaulin based system, start with Singi and Koi. And one was is 8 months in Singi and Koi in 4 months, because we understand the Vietnam Koi cycle is very short, 4 to 6 months, and Koi takes 6 to 8 months based on place to place and the water quality management along with the temperature. If I go to the next, if you see the cost of it 55 rupees, the growth rate is 120 kg in 5 months for Singi and Koi, 70 kg in 2 months. The approx price, sale price is 70,000, exclusive fish consumption. He already consumed 50 kg fish himself also, because when we have live fish at home, whenever somebody comes from outside, we want to serve them also fish. So that 50 kg also I take into consideration. So 3 to 5% mortality is there, but still he got very good success rate in the productions and the economics in terms. Srikant Dash. And here I will show you the Mr. Gulio. He tried to culture, is a Tangra fish. He's got very good market here, and with common carp. So in biofloc, many people talk about monoculture of species. Many people talk about single species cropping cycle. But unless and until we know the system, we can do polyculture based of system. Even we can do Vietnam, Singi, Vietnam koi, Desi Magu, Tangra, those kind of combination, even with common carp, with Puntias japonicas, Japani putis. So those kind of fish also we can understand, we can add, because we need to understand one fish, like a fish physiology along with the fish biology and their food compatibility. Once we know it, we can address it. So here, if you see the FCR, most of the cases, if you see the FCR is 0.9 to 1 or 1.1, but in the pond-based system, if you go for this kind of culture system, it's always is 1.2 to 1.3 and much higher. So he's not yet sold, though there is some mortality issues is there with air pumps and all. So always try to understand, because many of the speaker here, they have always addressed two things: good water quality, good aeration system, good water quality management can lead to good fish productions and good size of fish at the last. So I like to show you some of the backyard fish farming, what is going on in the many places. Aquadoctor solution is not only considering the, the commercial scale of the farming, and our aim is to take fish farming door to door. So every people, the household activities, they can be engaged with fish farming for their livelihood along with the as an income source or part-time. So this is a farm, and these projects we are now doing in home-based system in Tripura. Cemented tank also system we are propagating. Water quality management is very important along with the floc management. Those parts also need to be taken care while doing it. We are also trying to, small scale biofloc unit, portable, foldable tanks also we are now promoting for rooftop. This one I want to show. Many people talk about with a very big investment or the very big farm. If you have a very small land where you cannot not growing any kind of things, you are not using that land, just dug it out, put a simple polythene sheets, make the water stored. And even we understand in the northeastern states, though northeastern states is very hilly region, we cannot get every places perennial water source. So we are trying to get the rainwater harvesting process, keep the water, make it in a such a way that that we can do the perennial fish farming, not the seasonal event. And this kind of ponds we are now trying to do, like we are buying seeds, small seeds we are growing in this system, and the bigger size of seeds from this tank we are trying to shifting into our bigger tanks where we are making tarpaulin tanks or maybe the cemented tanks. So those people, I saw lots of questions, people ask for seed nursery management, fry rearing, fingerling rearing, those kind of aspects. I will show you another photos. Now we are also working with many of the hatcheries where we want to work with the larviculture aspects of, where we are using commercial feed or maybe artemia or maybe the live feed enrichment process. That enrichment process can be done with biofloc system even. This is also in cemented tanks where we pasted the, tarpaulin itself inside the tanks. This is a hatchery projects in West Bengal. We are now working with them. The main concept is when the baby fish come out, their mouth is small, and our bacteria, as Professor Iram very clearly mentioned, very nutritious, size is very small, and the digestibility rate of a biofloc organism is much more higher than any commercial level of feed. So we are trying to inoculate and do weaning process in hatchery process like this. And now we try to say and sell farmers also that, see, the fish, what you are looking for your grow out culture, that fish we are trying to grow in a hatchery process of biofloc system. We understand that the baby, when they take the biofloc aggregates in their system, the baby itself, when they want to grow out in a bigger scale, the same food when they will get, they will have more likely to have it. So that weaning process we now started in the hatchery itself. This is a picture from Bangladesh. And we also try to give trainings and employment generation aspects. It can be done unless until give them hands training on scientific management of biofloc system. Fish farming cannot be done by seeing only YouTube or just listening to the lectures. Fish farming can only done when you are jump yourself inside the system, understand it, understand the fish biology, understand the fish ecosystem, understand the water quality management, then only you can understand the whole process of your cycle. Biofloc will be success unless and until we understand the whole process of management. And lots of people maybe understand that, okay, biofloc fish farming can be done by man-based systems, but no, we are trying to now employ our housewives, our women empowerment through biofloc fish farming, because biofloc fish farming is a process where we can start with a very small period of time we need. So the housewife, after doing all the household activities, they can also give 2 hours, 3 hours time in the day, in the afternoon, the evening in the tank management. Even this is a women self group with 45 members of members. They are doing biofloc fish farming for last 1 year, and they are now very happy. Now they have 4 units. Now they are trying to multiply into 10 units. So this is a system where we need to make people understand what they are going to do. And the cement and tank system, many people shows, and I want to show one picture. This is a oval kind of tank system, because many people, even Mr. Hitendra was talking about tank, system, how you have to make the round tanks and all. But what I have also understand and seen, oval-based system also gives better, water circulations and the uniformity of oxygen throughout the tank systems. Oval-based system also, we can just look out in the system in a such way that based on density of fish, what kind of system you want to address it. This is a tarpaulin tanks I like to show, because placing of air stones or maybe aero tubes is also depends on the way you want to place it. Sometimes people place it like this. Sometimes people place it 1 inch in the top. Sometimes people place it, few numbers they place it. But there should be always a condition. There should not be any dead zone inside your biofloc system. Because if there is a dead zone or a dead zone pockets are more, there will be a organic sludge accumulations, and the bacteria, bacteria accumulation rate will be also there, and the fermentations or maybe the ognoxious gases, the ammonia production rate will be much more higher. So that's the important. And these are the, now we are also trying during our COVID system, like a COVID period, what the pandemic is going on throughout the worldwide. Now, many of the college going students, now they are at home. So we are trying to bring them out also in this new venture. These are the college going students of second years. We are also trying to give them training and make them a small group, so that these college going students also, while studying, they can also earn some sort of money through coming up this biofloc system. And I tried myself to put the ideas such a way that, biofloc is a system. It's need your determination, your time of understanding, and your dedication towards the system. Because fish farming will be success unless and until we go more inside to it. Fish farming will give you more, food security or employment generation unless and until we know it. So thanks for be patient with me, and thanks for listening the presentation. Thank you so much.
Thank you, Dr. Debthanu Burman. I see a lot of energy and only channelized towards Northeast region. Kindly extend your energy and knowledge to the entire nation. We are with you. We will provide you the place, lab, everything. Many farmers in Karnataka are looking for your services. We'll come back to you with some more details.
Sir, sure. Thank you, sir.
May I now request Mr. Vishwanath, Senior Assistant Director of Fisheries from Department of Fisheries, to share the schemes and initiatives of the Government of India under the scheme Pradhan Mantri Matsya Sampada Yojana and the schemes of the Department of Fisheries. Mr. Vishwanath, please. Down. Mr. Vishwanath, are you there?
Yes. Good afternoon.
Yeah, please. Little louder, please.
Yes. Good afternoon, everyone. Thank you for sharing the floor. I am here to share my views from the government perspective. We so far audience have heard a lot about the biofloc technology from the father of biofloc and various experienced farmers, entrepreneurs have shared their views. But our job is to manage this technology in what is coming in. As a government, there is a lot of gap we recognize in our own understanding of the technology and capacity building of not only the farmers, but also the officials of the department across India, and not only in Karnataka, I can say. There is a critical gap. We need to pick up quickly and come up with solutions to bridge this gap. We thank Dr. Magda for organizing this webinar, and especially this sort of webinars will, in the meantime, will try to fill up those gaps. So second important thing we are talking about is Matsya Sampada Yojana. Lot of focus is being gained to this particular schemes and program. So from 15 to 19, we handled Blue Revolution schemes. And now we are shifting to Pradhan Mantri Matsya Sampada Yojana. Many are saying it is a second version of Blue Revolution scheme. But I differ from their views because there are a lot of differences I would like to quote here. One from Blue Revolution was sector specific activities were focused, like inland fisheries, brackish water, marine, like that. Funds were flowing in some post harvest. But there is a shift in the focus in Pradhan Mantri Matsya Sampada Yojana towards activity based programs like production and productivity oriented schemes, technology infusion, infrastructure and post harvest management, fisheries management, and regulation. So the activities are gaining focus rather than the specific sectors. Of course, the sectors are part of those activities. So when we see the difference, another difference is from Blue Revolution schemes moving to Matsya Sampada Yojana is we are shifting our focus from production from the mass to the mass production. So because I am saying this because the production targets was there in Blue Revolution. The fund flow was around 3,500 to 4,000 crores. Now suddenly the quantum of funds has been increased, of course, including the beneficiary contributions, and is to increase to the tune of 20,000 crores for the same period, say 5 years. So what should be our capabilities to absorb, not only from the government side, from the entrepreneur side? Are we ready to take up this challenge and move ahead? The second difference is the technology. Again, the components that are being introduced into the Matsya Sampada Yojana compared to Blue Revolution schemes. Technology infusion is one of the major components which we are focusing to bring in more technology to increase the production. The production targets, I would like to update what Dr. Magada said. It is present production is around 14.5 million metric tons, and the target for the next 5 years is 20 million metric tons, which again there is a difference between Blue Revolution to Matsya Sampada. The targets are being clearly communicated to all the states. For example, Karnataka has a target of producing 1.2 million, lakh tons, say 0.12 million tons in the next 5 years. These targets have been communicated to each state so that the focus and the attention of the states are dragged into the schemes. Now the structure of the schemes, operationalized guidelines of the schemes have also been released. A lot of, can you hear us? Okay. So a lot of attention is being drawn towards these targets from the states. The number of meetings and the number of activities going on compared to previous 5 years is increasing day by day. So a lot of focus, again, the policy makers, the media attention has also increased on Matsya Sampada Yojana compared to Blue Revolution. So there is a lot of responsibility on every one of us to bring these objectives of Matsya Sampada to reality. So second thing I would like to tell about is the technology infusion schemes, very specific to this forum. There are 5 major components, mainly for the technology oriented schemes. Mainly for biofloc, there are 2 components, one for freshwater, another for brackish water saline, 14 lakh per 0.1 hectare unit, and for 18 lakhs for 0.1 hectare unit in case of brackish water. So there is RAS components for different types from 7.5 lakh unit cost for backyard unit to 50 lakh to the larger scale unit. So Government of India is also coming up with the different kinds of guidelines, the different set of guidelines for entrepreneurs. So it is yet to be, it is yet to come, and they are informing the states to reserve at least 15% for entrepreneurship model programs, just to encourage entrepreneurs with different kinds of technology integration, integrated models. Here, the basic difference in the existing guidelines is the flexibility of operation. The specific components, units have to be followed as per the unit cost specified as per the components specified in the guidelines. But in the entrepreneurship model, as we heard, there will be a lot of flexibility for the entrepreneurs to make their own projects with the combination of post-harvest or production systems and other activities. So this is another major change we see from Blue Revolution schemes to the Matsya Sampada Yojana. So there are a lot of things coming up. We need to identify the gaps within us to absorb this kind of funds. Main requirements I would like to specify is the technology value chain management. Where are the inputs available for each technology? Because Karnataka itself, now we have received more than nearly 200 applications for biofloc RAS applications. Many questions will come from where do we source these equipments? Where do we source these inputs? How do we bring the cost of production down? These are the questions we need to work on in the future if we need to stabilize this activity and take it forward and make it sustainable. And second most important, many of the speakers highlighted, is the production cost. So here there's the mass production is the goal. So the volume is the key here, not the, therefore the production cost and the margin of the profit is very less. So the entire profit depends on the volume that we produce and the little margin that they may get. So if we do not focus properly on the cost of production, what is the input, what are the input costs? So again, these technologies will not going to be remain for a long period of time unless the people involved, entrepreneurs involved, focus on the cost of production and the production marketability of the margin of the profit. And the third most important requirement is the absorption of the increased production. And are we ready with the kind of post-harvest supply management that is required to manage this production, increased production that we are anticipating through this technology infusion? So these are the some of the thoughts I would wanted to share in this forum. Thank you very much, Dr. Mukada, for giving me this opportunity, and also I thank all the organizers. Thank you, everyone. Thank you.
Thank you, Mr. Vishwanath. Participants, please go through the websites of Department of Fisheries, Government of Karnataka, or any department in your state, Fisheries Department of your state, and National Fisheries Development Board. You will get to know more about Pradhan Mantri Matsya Sampada Yojana. And now we come to the end of presentations. Now it is open house discussion. Kindly participants, be precise in your questions, no repetitions of questions, no counter argument. You may disagree with some of the points. Agree to disagree is always there, no counter argument. Be polite. And first I'll take on this question. It is in the chat box. I have answered this question. However, for the benefit of all the participants, I want one of the resource persons, like I want Mr. Hitendra, because he is from the chemistry background, and Dr. Deptanu to answer this question. What is the functional difference between biofloc technology and probiotic system? First, I like to ask Mr. Hitendra.
Could you please repeat the question, sir?
Yes. What is the functional difference between biofloc technology and the probiotic system?
See, in the biofloc technology, right, we are initially we are going to inoculate a couple of good
Probiotic.
bacteria.
Okay.
Yeah, probiotic. It's called probiotic because it helps us. It's a pro. It's
system.
pro. It is pro towards the good I mean
Production.
production and maintaining the water quality and all this. So initially we inoculate some bacteria, and we see to that over the period of time they can clear, I mean, multiply and you know and sort of take over the system, wherein the toxins are continuously reduced. So the carbon is added in such a way that this the nitrogenous waste is absorbed by the bacterial cells. More number of cells are produced, and then these because these bacterial cells, these are single cell protein. Single cell protein is highly digestible. So fish can easily, the fish of different age groups, even the fingerlings, the small ones, all of these can easily consume the single cell protein. Okay, and the protein assimilation is also very high because it's a single cell protein. So probiotic is just the initial additive that is needed, because if you want the flock to happen as such, it will happen over a period of time. It will happen. Okay, but that will take more time, and because these are commercial operations, you need to stabilize the system as quickly as possible. So that's the reason we infuse probiotics in the beginning, and then try to maintain the system. So and the other aspect, what was the other thing that you asked, sir? Probiotic and
exclusive probiotic system and the biofloc.
Oh, see, in biofloc, because see, the one thing is we are separating the water from the soil. We are keeping the soil away in terms of biofloc. Whether you use any of these HTP liners or tarpaulins or any of these, you keep the soil away so that you don't have sludge getting into the soil and and fermenting. I mean, anaerobic fermentation. Yes, for anaerobic fermentation and all happening. So there also, when they do Vanami culture and all, they continuously add probiotics. There's a regular addition of probiotics. I mean, every 3 days, 1 week, depending upon the densities of system. So in biofloc, you don't have to continuously add. You don't have to continuously maintain, because your good bacteria are killed by the Vibrio and all this in mud conditions where soil is allowed to interact with water. But in biofloc, we tend to block this interaction by using a sterile medium, HTP or tarpaulin, whatever. You try to block this interaction and only give opportunity for these good bacteria to continuously develop in the system. So that's the main difference as far as I've understood.
Thank you very much. Very apt and precise. Dr. Depton, you want to add anything more? What is not said by the Hitendra?
I just like to give one real life example. Now in COVID situation, before we eat, we do lots of sanitation and hand washing. But when COVID was not there, just washing with the simple water, we start eating. Biofloc system, just consider is a COVID situation. And whatever we're doing in the pond farming is a normal practice of probiotic adding, getting some result, and just doing it. In pond system, what we have observed, there is a lots of bacteria interaction, and always there is a unbeneficial bacteria growth is much more higher because the sludge content is much more higher with lots of organic matters, which we are not controlling in the situation. But in biofloc system, we control all the water parameters side by side. Addition of carbon, only objective is our heterotrophic bacteria should get multiplied and the organic waste should convert it into microbial protein. That's what is a simple example of what I want to say, sir.
Thank you. Another question from one of our participants: can we treat the diseases with chemical agents in the biofloc system?
I just like to add one simple thing. Now lots of diseases come. I think even ICR, all the research institute, even NVFGR also, I address that issues. In Vietnam Khoi, when there is a high density farming is there, lots of disease come, and people say different type of disease names, the viral, bacterial, lots of research needs to be done. But when the disease come, they use lots of sanitation and other stuff. But I think always in the biofloc system, we need to improve some sort of feed supplement which improve the immunoboosting kind of stuff. And if there is a disease is there, maybe small dose of water sanitation process we can go through. Because if we use lots of high amount of sanitizer, say example, I like mentioned, like people use commonly BKC or maybe glucaraldehyde or maybe formaldehyde or maybe iodine solution. When we use a huge amount, then our beneficial and unbeneficial both bacteria are die. Thus again we have to start with FCO preparation or the bacteria preparation kind of stuff.
Professor Yoram, your comments, please.
Okay.
Yeah.
As to the probiotics, probiotics, some of them are very good, especially in very small systems like nursery, not in open ponds that hold for a long time. Because the natural population takes off. In case you have a biofloc system with a variety of organisms, the probiotics do not add anything to the protection of the fish against disease. So when you have a biofloc system, usually you don't need to add probiotics.
Dr. Babita. Yeah.
Yeah, there are certain farmers who are using only the probiotics for their culture system. But in our experience, what we were doing the pond soil based inocular method only, and we have done the genomic analysis of microbial diversity also. So we have found so many organisms, huge number of organisms. In the commercial probiotic, we will be able to get 6 strains, maximum of 10 strains, not more than that. But in this case, actual bioflow contains huge biodiversity of organisms which are having various functions, which varies from suppression of pathogenic bacteria to lipid metabolism, carbohydrate metabolism. Everything is involved. Even we have studied the functional metabolism also. So it has given me a huge concept that these all bacteria are required to sustain the system in a very, very good way. So that is how we are managing the commercial system. We have not used any probiotic.
Many people have asked about the disease management. One general understanding is in biofloc systems of 10 ton to 30 ton, 40 ton capacity tanks, we can manage the entire water quality. So we can, if you want to exchange the water, you can exchange the water entirely. So management of disease, crop, everything is manageable. So in that context, answer is yes, all diseases can be managed with the allowed chemical agents, and water exchange will subside the disease by many folds. So now, very basic question people have asked: how to maintain unmuted Pratibha, Babita Rani, how to maintain CN ratio. So friends, this is very simple question. There is a thumb rule technique, but it all depends on the management. The principles of aquaculture cannot be written in indelible ink. It all depends on your resource type, management strategy, stocking density. So there are umpteen number of parameters which will decide the production system. But in a moderately stocked, like 300 to 400 numbers in nursery and 100 numbers in growth, the CN ratio can be maintained. Suppose if you have 100 kgs of fish biomass, and if you are feeding at the rate of 4%, you need 4 kgs of feed, right? 100 kgs of biomass, 4% of feed means 4 kgs. Normally, we feed the fishes with, sorry, 28 to 30% protein diet. So if we are feeding the 30% protein diet, the 4 kgs of feed will have 1440 grams of protein. So protein will have 16% of nitrogen. It is about 190 grams of nitrogen we have in the feed, 4 kgs of feed. So normally fishes are poikilothermic organisms, and they utilize 25% of the nitrogen and excrete around 75% of the nitrogenous compounds. So it is about 144 grams of nitrogen. So in order to maintain 10:1, you need to add 10 times of carbon source. It means 1440 grams of carbon source, be it molasses, sugar, soya meal, whatever it is. This all simple rule, but you cannot stick to the thumb rule technique. It is completely based on the observation and the situation of that particular day, depending on the region. Okay, we'll come back to you with more details. And can BFT tanks, biofloc technology tanks, can be used as RAS, Recirculatory Aquaculture System? Yoram, please, Professor Yoram. Can biofloc technology can be used as RAS?
There are several. Well, first of all, biofloc and RAS are different systems with different philosophy. In RAS, the philosophy is don't let any residue in the water. Flush the water continuously, filter it, and have super clear water in the tank. In biofloc, we don't want super clear water. We want water with many bacteria. However, there are possibilities to combine, to have a system that has limited number of bacteria, not very many bacteria, and to have in the side a tank that will take care of the water and will recirculate it. So there are some possible combinations between RAS and biofloc technology. There are now recently some papers published about this, and it's possible. It's an interesting approach, and I assume that in the near future it will develop.
Thank you very much, Professor Yoram. Very recently, myself and Mr. Ramesh, who is aquaculture consultant, we worked on the 100-ton capacity project of biofloc technology, where you need 65 tons of sugar or molasses. So looking at the huge requirement, we have to come out with some alternatives or some other combinations. Maybe at the zonal stage, as Mr. Hitendra and Ramesh said, it is not one-time stocking and one-time harvesting. If you can have three systems like larval rearing, zonal rearing, and growth, you can manage the biofloc systems in a better way. So at the early stage, you require the biofloc requirement is very less and management is easier. When you get along during the last quarter of the growth, the requirement of carbon source is very huge. If you can rightly manage autotrophic food chain, heterotrophic food chain, and the little water exchange, and with good protein diets, you can cut down the cost on production. So that's what, more than the science, you need to understand the economics and situation specific management strategy you have to come out with, because it is the cost of feed in Northeast region is very costly. In some part of the state, it's very cheap. In some part of the country, in our country, I'm telling, the cost of electricity is 2 rupees in some states, but whereas in some states, it's very huge. So in that context, this all depends on the management. You have to come out with right management package for each of the specific sites, be it number of tanks, the species. Very recently, Mr. Ramesh said, we can even grow scampi in the biofloc system as a service species. Even if you stock 5,000 numbers in a growth tank, even if you get 30 to 40 kilos of, not scampi, I'm sorry, the venami, that will take care of the labor cost. This is how, and Dr. Devtanu said, we can combine many of the species which can compatibly live and which can accept the commercial diets. If they are not cannibalistic each other, we can grow any fish species and we can have a staggered uh, harvest. So definitely we can grow pabda, marl, koi, because they fetch very good price in the market instead of growing the same species like tilapia, paku. So definitely we have to work out on the stocking density and right species combination and come out with the right package. And definitely our university is ready to join hands with the entrepreneurs, farmers to understand the science part of it. Now I want to listen from Dr. C.K. Murthy sir. He has worked in National Fishery Development Board at the capacity of Executive Director. I understand Dr. Ergal is also there online. We want to hear from Dr. C.K. Murthy. After that, we want to hear from Dr. K.N. Prabhudeva, who has worked on saline and alkaline soils extensively in Karnataka. Dr. C.K. Murthy, sir, please.
Thank you. Thank you, Dr. Shukumar. I think we have heard for a long time various situations of biofloc, starting from the basic concept up to the practical field. I may not like to add too many things, but only from the commercial aspect of it, from the farmer's point of view, especially from the Indian context, what I have seen is that the biofloc system working really well for tilapia and for shrimp, Vanami. Places where tilapia has got good demand, like Kerala, Tamil Nadu, and the northeastern few states, probably the biofloc system is really profitable. As far as when we come to Bangalore, Karnataka, in and around a metropolitan city like Bangalore, Mysore, or major cities, your marketing, we are finding that marketing is becoming relatively easy and they can get a better price relatively, so that this becomes an economic viability. So my request is that to all the entrepreneurs, with all the advertisements, what we are hearing about Prime Minister's Matsya Sampada Yojana and other schemes, be cautious about the economic part unless you are sure about the cost realization. Now this with a high electricity cost in some of the states like Karnataka, even Andhra Pradesh, you have to be little careful about the system. Okay, so those 2 points, one is economical aspect of it, you have to be extremely careful before venturing into biofloc system. Now second part is that seed rearing part, across the globe, lot of people have shown good results, but lot of standardization is required. What is the size, what is the stocking density, the feeding rate, and other things. So I request some of the central institutes like now CIFE is doing work. So this has to be done on a large scale basis, because this can be a very good profitable avenue for farmers to get revenue for the better seed for the culture system by adopting the biofloc, which is in a rearing area. As we know, the constraints of rearing area is one of the major drawback in fish seed availability in many states, except maybe West Bengal and Andhra Pradesh. So in that way, that kind of package of practices, instead of giving tidbits, if we can give a clear package, what should be the ideal size and what should be the sucking density and things like that. Third issue I just want to highlight is that some of the people are advocating backyard hatcheries. Now please suggest whether it is really economically viable for the farmers to go with a very small scale, say 4-meter dia ponds, 5-meter dia ponds, considering the input cost as well as the production. Now, unless it is economically viable, that farmers may run into loss, and also that will bring a lot of bad name for the system as such in the future time. So be careful and be scientific, and economic aspect also be take into consideration before suggesting these 2 things. I think that's all the points I wanted to share. I will thank Dr. Shivkumar for giving me an opportunity to share my views. Thank you.
Thank you very much, sir. As Dr. Murthy sir said, many gadgets producers are propping up in India, like plastic lining producers, blower producers. But on the other hand, we don't see a spurt growth in terms of seed production of different species. So like many of the biofloc farmers, they have to join together, form a farmers producer organization, and they have to start their own hatcheries instead of depending on somebody for seed and waiting for many years. They should be able to produce their own seed requirement. I see Dr. Azad sir from Kuwait. Dr. Azad Ismail Saib is working on breeding of pomfret. He has joined us from Kuwait. I want to hear from Dr. Azad sir.
Thank you,
Azad
Dr.
sir,
Shivakumar. Yeah,
yes,
yeah.
sir. Acha.
Thank you for giving me the opportunity. It's nice to listen to Dr. Yoram and many of the friends from India. Debantu, he's from Tripura, I believe, and Hitendra, Dr. Babita. It's very nice to hear to them all. Dr. C.K. Murthy is there, my senior, my friend, and Dr. Vishwanath was also here. I have some pertinent questions related to conditions here in the Arabian Gulf and all those things. Of course, here the biofloc technology is yet to pick up. Probably Saudi has started to some extent, and in Kuwait we are with one army in biofloc system. It's only in the beginning. I have one question to Professor Yaram. You said about the floc nature of the floc. It varies with the aeration systems. Different aeration systems. You are talking about different kinds of aeration systems. How to decide about the quality of the floc and when the sludge has to be removed? Is there any parameter that we can assess? And number two, Dr. Yoramundi, here the Arabian Gulf region is marine or supermarine. Like most of the times it is more than 40 ppt. And in Kuwait it is 44, 45 ppt. And it's almost at the fag end of the Arabian Gulf. So what kind of biofloc system you suggest? Is it worth taking biofloc system in these kind of situations? So this is what I have in mind for asking you your clarification. Thank you.
You can speak, Professor Yoram.
Okay. Well, first of all, the two last questions. One is the control of biofloc. There is a lot of effort on being an ideal composition of bioflocs. Some very interesting work in Australia, almost to commercial. It got to commercial application. By the way, most of the bioflocs are added to the feed, so you have a controlled supply of some kind of organisms in the feed, and it increases the quality. It's very difficult to change the composition of a complex system like a pond that has to for a few months is stable. But there is some work done on it. In the next few years we'll have better idea. Regarding the salinity, the 40 to 55% salinity in hot areas like Kuwait, problem is not the resistance of the bacteria to the salinity, but it is the resistance when to salinity of above 40 ppt, the shrimp does not grow as well, so the limitation, not the bacteria. I would like to add another comment. There were big discussions about the price of feed and the effect of the type and the feed on the economy of the production, which is very, very true. If you understand the biofloc system, you can save in feed. For example, if people use for fish, uh, feed with 30, there are a lot of nitrogen is accumulated in the water. You have to put a lot of molasses to keep the protein down, and you get to a cycle where you waste money on feed and you waste money on a lot of molasses. However, instead of doing this, you can feed the fish not with 30, 35% protein, but with 20, 23% protein. Then there is less release of ammonia to the water. The small amount of molasses or whatever, rice bread, etc., can take care of this nitrogen. So you save on feed. You don't use too much molasses or others carbon, and you don't have an excessive concentration of biofloc system. So you have to control the system in a way as minimize protein, minimize feed. You can have less feed when you have biofloc system, and you can have less carbon and less money and better income. So the idea is you have to control the whole system. By the way, as to the comment of Dr. Shiva Kumar, having a few steps in the growth, the nursery step, and later on is very, very good. And it has to do with the biofloc. For example, the works that we heard Kenya, it was that if you have a nursery and you have actually you grow in the you grow this zooplankton that can be used, the small fingerlings, they will grow better, you save on expenses of an artificial feed, and then you be different when the fish are larger, need another stage, bigger ponds, you get the water better, so having stages is a very, very good as
Thank you, Professor Yoram. There's a question from Mr. Vishwanath from the Department of Fisheries. He's a Senior Assistant Director of Fisheries. He is asking the team to develop a standard operation package, technology package for biofloc technology. Can we join together, all of us, Mr. Devtanu, Professor Yoram and Hitendra, and many others who have joined this webinar? Can we come out with a standard package of practice so that farmers can take up without any hiccups, and they can follow some of the existing practiced procedures? So I'll invite and request their inputs. Mr. Tendra, Devtanu, and Yoram has already agreed to be a part of this mission to augment fish production in India to attain 15 million tons of fish by 2025. And ಇಲ್ಲಿ ಸ್ಟಾಲಿನ್ ಫ್ರಾನ್ಸಿಸ್ ಅವರು ಹೇಳಿದ್ದಾರೆ, ಕನ್ನಡದಲ್ಲಿ ಇದ್ರೆ ಚೆನ್ನಾಗಿರ್ತಿತ್ತು ಅಂತ. ಅಂಡ್ ಪಾರ್ಟಿಸಿಪೆಂಟ್ಸ್ ಕ್ಯಾನ್ ಆಸ್ಕ್ ಕ್ವಶನ್ಸ್ ಇನ್ ಮಲಯಾಳಂ, ಕನ್ನಡ, ತೆಲುಗು, ಹಿಂದಿ, Urdu. I think Azad sir can speak in Urdu, and Devtanu can speak 5-6 languages, Hindi, and any language you can ask, and somebody will translate it. And it is pan-India program, and the participants from 17 countries are watching this and very happy to share this information of the demography of the participants. And it's a very huge response from all over the country and other parts of the world. And most of them are watching in the YouTube. And kindly raise your hands. I see a lot of hands raised. We can ask question in any language of India. I will try to answer it. ಫ್ರಾನ್ಸಿಸ್ ಅವರೇ, ನಾವು ಅಚ್ಚ ಕನ್ನಡಿಗರೇ, ಕನ್ನಡ ಬರಹಗಾರರು. ಬಾಲು ಅವರು ಕೈ ಎತ್ತಿದ್ದಾರೆ. Is he, I don't know where is he from. Please ask the question and somebody translate it, and kindly let us know with whom you expect the answer, so that we can direct the question to the right resource person. If it is general, yes, Balu.
Yeah, I'm Vidu Balu from Tamil Nadu.
Yes, sir.
Yeah, I'm doing a biofloc with organic method.
Organic biofloc. Okay.
Yes, organic biofloc. And what you are all telling, it's all based on so many countries' research and all. But here in Tamil Nadu, we are doing in organic method, traditional method. We are not using anything.
Hitendra and Devtanu are from India and doing many projects in India.
Yeah. I now only I saw those projects.
Okay.
The same projects we have started. Even I started doing on the top floor of an apartment.
Very nice.
Yeah, because they asked, they want a fish to their own family. So I put a project on their top floor. So like that projects are all going to start. So many projects are going to start, but my project is like totally on organic, not based on any probiotics and all. Even a probiotic, I created on my own probiotics, because we can't buy probiotics. We can produce our own probiotics and
Mr.
carbon.
Balu, if it is not patented, please kindly share the details of your package.
Yeah,
we will spread it to the entire nation. We are very happy.
Yeah. That is what we are on some research. Even I am going to come to Bangalore regarding this, because of the code I'm stuck on that. The past 6, 7, 8 months I am working on that, and it's working. It's working, and death rate is very low. So many things I'm going to after finishing that research. I need your all help.
We are with you, Mr. Balu. Let us join hands together, understand the science, biofloc science, in some more details, and spread the proven technology to the entire nation. And we want entrepreneurs like you to spread the knowledge, make money, learn science, spread it. Thank you very much. And earlier slogan was, wherever water, let there be fish. Now, Devtanu and Balu, you have already started the biofloc tanks on the rooftop, on the roof of the houses. So wherever the space, let there be biofloc tank.
Yeah. That is what. This is a new project for me.
At least if they can produce the fish for their own. Now the
that is what.
The average consumption of fish in India is about 11.5 kilos. But as against the World Health Organization's recommendation of 20.3 kgs for healthy adult. So the gap is huge. It might take 8 to 10 years. So if we can promote the backyard biofloc technology, so that people can produce the fish for their own in the organic format, and if they want to make money,
that is what we are all planning for that.
Eat fish for health, grow fish for what? Eat fish for health, grow fish for
money,
wealth.
Yeah, wealth, health and wealth.
So thank you. I see Mr. Jagadish Ningappa. He was doing.
Thank you, Suprita for joining this session.
Thank you, Mr. Balu. And I see Jagadish Ningappa. Yes. Please lower your hand. Go ahead. Jagdish.
Okay, Dr. Magadha, excellent webinar. First of all, thank you. and for all your effort, congratulations.
Thank you.
And my question is completely non-technical, and it goes to Dr. Yugat. Basically, I would like to understand.
To whom? To Yoram?
Hello.
Question to whom, Mr. Jagdish?
Technical people, even Deptanu can take it. I'm fine.
Okay, Deptanu, please. Yes.
Okay. So this is on
And
the
Hitendra, please.
See, if it's for a first-time farmer, how would he differentiate the biofloc bacteria versus the sludge inside the tank?
Okay, Jagdish sir, very good afternoon. I will tell you, like when you start biofloc fish farming in India, many people always just say, whoever giving training, they are earning money. Whoever selling product, they are earning money. Nobody is talking about the technology, how they can understand. And now today I can see Shiv Kumar sir arranging such a program, and many people throughout the country is watching this program particularly. Now we are here for to know the knowledge.
Yep.
And the question what you ask, the question is still farmer asks me. Like, the question what you ask me now, farmer is still question asks me when I show in EMOF code, they're telling all are fish toilet maybe. How you are understanding this is bacteria and how they are moving. So in naked eyes, we cannot make it more differentiate. I say, boss, if you want to see your own tanks, I have a small field microscope. I can show some things which is live and moving. And if it is a toilet, when the issues come, the toilet things will not be moving. It should be settled down. So as a farmer point, we need to make our system also very farmer friendly. Today in India, whoever doing in biofloc fish farming, many people say biofloc is a flop. No, floc is flop. And I address one point, try to understand. See, each fish, some people here ask a question for a nursery management, what will be the water level? As we understand, for a small baby, they cannot eat in a bigger plate like us, right? Like an adult. For a small baby, we have to have less water volume in the tank so that my baby should grow slowly. They can look for the feed, they can eat properly, proper aeration should be there, and gradually we can increase the water level when we want to make it a grow out structure. That's what, sir, I can address you, because I also don't know what I have to say, but this is the way we can understand the farm level.
No, the reason is, see, I'm familiar with RAS, where you can easily remove all the debris, right, and make the tank look super clean.
Yes, very true.
But in the biofloc, it is not the case.
That's
right?
why. That's why.
Can I come in on this?
Yeah, that's why Professor Sir also mentioned is a suspended solid of many things together. Okay, understanding segregate even nematodes with probiotics or maybe bacteria with some other protozoa is very tough, unless until you take it into lab level of scale.
That's precisely, this has been such a big learning for me in this session. I was of so many of my doubts got clarified, today. But still, I would like to ask some dumb questions to get better clarified. In a biofloc tank, if I look at it, the watercolor would be very different from an RAS tank or an RAS system, right? So my basic question is, if I don't differentiate what is the sludge and what is the bacteria in the floc, how efficiently my system design I could make so that I can remove all the sludge and not the, the floc.
Can I come on this? Can I
go ahead? Anybody?
Yes, sir. I mean, if you're talking about the floc, right? It's an aggregate. So in the floc itself, there is some amount of the sludge matter, which is around the sludge matter is where the bacteria or all other algae or protozoas aggregate. So it's a source of they are trying to break down this sludge as well. So earlier this was actually called as ODAS. In the 1980s and all, this was called as ODAS. So the full form is organic detrital algae soup. So detrital is the waste, whatever the sludge and whatever the fecal matter is there, right? Or the waste feed or whatever is there, right? So the microorganisms, they aggregate around this particle and slowly they break it down. So that's how this works. So there should be some amount of sludge which has to be suspended. The heavier particles of the sludge will settle down because of centrifugal force, or because of the whatever, the density of that matter. It will settle down and you flush it out. So you want only a part of the detrital matter, or the fecal matter, or the waste feed to be suspended, so that the bacteria can do its job. The bacteria and all the protozoas and the algae and all can, they can break it down.
Thank you. Dr. Yoram, any non-technical response to my question?
First of all, I think that the difference between bacteria and sludge is not biological. The same bacteria that are very good, keeping the good quality of the water and the health of the fish, if they will settle down and accumulate in a place where there is no oxygen, they will start rotting, and then the same bacteria can produce in this environment with no oxygen, they can produce toxic elements. Right. So it's a physical management of the pond. If you aerate properly, if you drain the excessive sludge properly, then you have less sludge and more active bacteria. By the way, you can do it also with vertical substrates when you absorb the bacteria in the water and don't in the surface, don't let them settle down. So it's a physical difference, not a biological difference. It's a management issue. You can minimize your sludge if you properly manage the pond.
Mr. Jagadish, I told you in the beginning, 60% of the food in the aquatic system is less than 60 micron, 60. Remember this. Even fishes cannot harvest it. If you have played in the streams when you are young, or even now, go to any stream, you see the submerged aquatic weeds. Lot of fishes will be moving around the substrates, because these the smallest microorganisms and other diatoms, plankton, zooplankton, they assemble on the substrates. The fishes will nibble on that. Here in this technology, using right combination of carbon and nitrogen, all this matter can be flocculated, and that will act as a feed. In fact, in locally formulated feed, we used to add pig dung, cow dung, because omnivore fishes, they eat the mud and digest whatever they can and excrete the other things. So this is how the lower order animals will exploit the nature in order to obtain more protein from the system. But we are enhancing it by maintaining carbon and nitrogen ratio. So by managing it, ultimately it boils down to nitrogen, phosphorus, K. So this is how the entire the waste can act as a feed that will bring down the cost on feed, external feed. This is the basic principle. Ultimately, we want to reduce the cost of production by exploiting these excretes and other nitrogenous compounds.
Okay. Thank you so much.
Yeah, I agree.
Who is this? Ashok Arman Saeed. Are you from Delhi, Arman? Unmute.
Yes, I hope you guys can hear me now.
Yes.
Yes, I'm from Delhi.
Yes, please.
Yes, so so my question is, since I'm a beginner at this, and apologies in advance if it's a very primary question, but I think also Ditanu will experience it because he is in the upper region of India, where we do experience very harsh winters, like we do in Delhi as well. Okay. Now, during these harsh winters, because I'm guessing we'll have 2 to 3, at max 3 harvesting seasons. And if we have about even 2 harvesting seasons, one is going to primarily fall within the winter season. Now, during the winter season, when we are having very low temperatures, how do we tackle this when we are going for a biofloc? Because even a smallest error or for a very small period of time will make a huge difference to our harvest.
We did some work on overwintering or growing of tilapia during the cold season. And then we did it by putting greenhouse plastic over the ponds, having high density of fish in this pond. And we could get to temperature just by putting normal agricultural plastic, could raise the water temperature by about 4 degrees. If you are in a real season, then you need some heating, and then economical question whether it pays to grow fish that or streams that need high temperature in the real cold places.
And
Mr. Arman, Mr. Arman, by putting shade nets and polyhouses, you can increase the temperature by 4 to 6 degrees. If it is extremes, there are induction water heaters. You can use them in the pond itself. If you don't want to use the induction water heaters in the biofloc system, you can have the normal reservoir tanks where you can heat the water. Such water can be used into the biofloc system. There are strategies. Temperature is not a serious issue. It's a manageable issue in the biofloc system. That is what the system is. We should be able to control everything, right from stocking density, water quality parameters, temperature, pH, everything. It is controllable. Yes, please. Yeah, that's what I said. It polyhouse, shade net, and reducing the water depth also increases the temperature. So, or increasing the water depth so that fishes can go down and hide themselves. So there are many strategies. We'll come back to you. And any other questions? Eric, we want to hear from you. You have a lot of field experiences. Dr. Eric, please. Tell us some field solutions, not the lab solutions.
Thank you, Dr. Magada. I'm happy to hear so many questions coming from participants. And indeed, we are getting
today no
right.
Food is delayed by 1 hour for better cause.
Yes,
please.
We are actually involved in some of the field experiences in terms of biofloc. And we have seen cases where farmers are asking so many questions on how they can be able to articulate this technology and understand and apply. That's why I did mention that we are going to partner with textile industries to be able to install pond-based biofloc technology in one of our counties here. And it's going to be one of the centers for learning also for the youth and other people who will want to get hands-on experience in the technology. We have also encouraged people to use the technology to produce zooplankton. We have developed rotiflocs and copperflocs, which are producing this zooplankton. And we have seen that some farmers have started to register good survival rates in their laviculture activities in the specific hatcheries. So we are glad to partner with other people. I'm happy that Eurom is also willing to partner with us, with everyone. So we are going to be able to work together in this regard. And going forward, you mentioned, Dr. Magada, you mentioned about the standard procedures. Maybe this is a very interesting thing to look at so that we can be able to have standard practice that can cut across in regards to biofloc technology implementation and adoption. Thank you very much.
Any questions from other people? Imam Su, are you there? Imam Su, yes.
Hello, sir.
You finished your lunch?
Am I audible? No, sir. No, sir. I'm waiting.
Okay, okay, okay. Please, please. You wanted to no contribute your experiences, please. You are also working in the Northeast region.
Sir, actually,
Please, please.
I must congratulate you, sir. I took the lead of this session. I know, sir, you are the leader when I was reading in Mangalore College, and you are reading this bonding issue. Sir, I have few doubts from the experts, sir. Though I have not any expertise in this biofloc, I have some expertise in that I came across some experience in RAS and the cage culture technology. I thought that biofloc system is a fully intensive farming system, and it is a mainly the nitrogen management system where we can converting the nitrogen to the microbial protein. Sir, I have some doubts from the those are the experts that first thing, whether this biofloc is now anywhere of the India in freshwater condition, is it successful? If it is successful in freshwater condition, then what are the species? Then if you come to the species, then the people in West Bengal, I am placing at West Bengal in Sifri, Barakpur, Kolkata, and people are going for the tilapia and the Vietnam koi. What I am seeing in the 2-3 biofloc farms, because it is not our mandate of the institute, but I have came across some of this biofloc system, and whenever we are giving training to the farmers, they are repeatedly asking the same thing. But sir, what happened reality in the ground, that when we are producing this tilapia or Vietnam koi, say the expert can say in better way, what is the cost of expenditure? We are, if we'll go for that BC ratio, we can say the BC ratio, the exact BC ratio that farmer can adapt. Second thing, if we are going for the tilapia and the Vietnam koi, in Kolkata market is the hub of the market, in Howrah fish market, there the farm, the tilapia is selling around 100 rupees or 120 rupees, and the farmer is getting maximum hardly 70 to 80 rupees per kg. Whether if the farmer will get the 70 to 80 rupees per kg, because in our country, what I am telling this in context of India, because if it is the Western country, it's some of the foreign countries in the middle part of the Saudi, there the tilapia will get the $6, $7 up to $10 per kg. But in India, that is actually really not happening. So if we'll go for the economics, so whether the farmer will get the profit from out of this freshwater condition, no doubt there is.
Please reserve your questions. We'll split into the smaller issues and answer one by one. You asked about the freshwater biofloc system. Is economically viable or not? My answer is yes. How you will prove it? Second one, the market dynamics, people are not, they have not changed their pricing policies, but you should be able to place your product in such a way that it fetches better price. So that is what, if you can form a farmers producers organization, you can come out with fillet, boneless, curry cut, and we have to work more on the marketing side, so that, see, I have seen in my own eyes, in Areca plate, farmers are cutting the tilapia fish in a shear fish manner, positioning it in the fish shape, put a polythene sheet and give a masala, and they are selling it for rupees 200. And Mr. Jagadish Ningappa, who is here in this webinar, he was selling the live fish for rupees 300 to 400 rupees. So if you can sell your produce, 30% or 40% of your produce with value addition, and even if you sell the rest of the produce for rupees, see, what is the bank interest? It is 8 to 12%. You know, the benefit cost ratio of paddy in the entire country is 1:1.2 in paddy if you get 25 quintals per cycle. Sugarcane, the benefit cost ratio is 1:1.8 in fish, whether it is extensive, modified extensive, it is anything more than 2. In biofloc, I have worked out the benefit cost ratio. It starts from 2.4 to 3.5. So I have seen many biofloc systems have failed because of the non-technical reasons and blaming the science and the scientific fraternity. If Israel can adopt this technology 2 decades ago, we are lagging behind to copy the good things. So that's what I see. A lot of promising issues. Of course, there are issues, something to do with market. So even if you get profit of 15% and above, it is more than the bank interest. In that context, I see aquaculture in any of the systems is profitable. To maximize your profit, you have to learn how to cut down the cost through science and technology. If Yoram can do it, if Eric can do it, we can also do it, because the principles of the science remains the same throughout the world. There is no separate science for Israel or the Eric. If you believe in the principles of science, we also should be able to produce the same output by following the same methodology. So I have answered your question. If you still have the doubt, when we prepare the standard operation procedure, send all your queries. We will put them in FAQs and we'll get the answer from the right people and solve those issues. Himanshu, still if you have a question.
Okay, no, sir. It's okay, fine, sir. Later,
No,
I will set.
I'm in a hurry because looking at
I
the
talk with my telephone, sir.
don't
I
think
talk
that
with telephone.
I'm happy with your questions. I'm in a hurry because
Sir, actually, it is yes, sir. I'll talk with later, sir.
And
Personally, I'll talk.
I just like to add.
Devtanu.
Yeah, I just like to add one thing to Himanshu sir. I'm also stay in West Bengal. In West Bengal, fish farmers, what they do, they just do Vietnam koi culture throughout the year. They understand like 4 months they can sell 30 gram to 40 gram. But in bioflog is a concept where based on your market location, consumer preference, do multiple species culture, multiple stocking. Then the harvesting of the production will much more level we can take it. Unless and until we make our farmers very trained with the technology, we always blame the technology, the benefit of the technology we cannot harness. Thank you, sir.
Kindly suggest any other species other than the tilapia and
Okay,
the Vietnam
okay.
koi.
Okay, I can tell you by number itself. One is Pabda, Desi Singi, Desi Magur, Tangra, then Marels, then you can do Puntias Japonicas, then we call this Puti, then you can do common carp, you can do Amur carp, even in carps. Many people say that carps we cannot do in bioflog, but
in the West Bengal?
Yes, in West Bengal.
Can you?
It is there in West Bengal. I will share the details to you. You can go and visit yourself, sir. No problem.
Can you say contact details of the seed producer of the Singi, Pabda, and those can produce the seed and supply in a large number?
Yes.
Mr. Debatsu and Himanshu, I have collected the seed producers of different species in the entire country. I'll share with you.
Yes, sir. The farmer actually really need this issue, sir. The farmer are really facing. They are not getting the seed in proper. They are getting the seed from the Bangladesh. They are not getting the seed from the India. So that unfortunately we have not a seed certification agency in India. These are some of the practical issues, sir. Okay, later I will discuss, sir. It is already too late, sir. Later I will discuss by telephone. Thank you.
I request Dr. Manjappa, head of FRAC Esargata, and Dr. Rajanna, KB Rajanna, look for the seed producers across the country, collect the entire database and upload it in all possible social media, so that instead of getting it from the Bangladesh, maybe Northeast region people are getting it from there, no issue. But identify the people who are producing such seeds in India and publicize it, so that let us connect the seed producers and the growers. I take only 2 questions now. In another 5 minutes, we are closing the webinar. Last 2 questions. Dr. Prabhudev, sir. I see Dr. Prabhudev. Prabhudev, sir. Dr. Prabhudev, sir, was Director of Extension of KVFSU, and he was professor and head of this Fisheries Research and Information Center. He has worked extensively on reclamation of saline and alkaline soils. Dr. Prabhudev, sir, your comments, please.
Hello.
Yes, sir.
Hello.
Audible, sir. Audible, sir.
Can you hear me?
Yes, sir.
Okay. Yeah. First of all, I congratulate Magad and his team for the wonderful. This is a very good opportunity for many of the people to enhance their knowledge on biotechnology, particularly biofloc technology. It's a baby in in India actually. Okay. This technology has got, it's very close to nature, and we should congratulate Dr. Aram for converting his concept into technology, certainly. And it's gaining excellent momentum all over the world because it's very close to the nature. We are not putting any it. But my only concern here is that in India, lot of people are producing YouTube. Okay. And lot of people are trying to copy and implement the things. That is where I am afraid, because this baby should not be died at the early stage. It should be born and grown up into adult. And that's the thing. Second thing is, Vishwanath said that there are about 200 applications from different people in Karnataka for biofloc and RAS. These are all the special technologies. We need some special kind of seeds. Okay, we have to take into consideration some seed production aspects also here, because you cannot do with carbs because they are very cheap, and we have to do something about it. And second thing is, India is a wonderful country with 80% of its climate is blessed with tropical environment, and with different. It has got a very good scope for fish culture, and we need to promote fish culture in a big way, especially using saline alkaline waterlogged areas. In Karnataka itself, there are about 6 lakh hectares such waterlogged areas in different command areas. We need to take and my only concern is that India's as such, Karnataka in particular, India as such is facing severe supply of seeds. Okay, we need to give much importance to seed production and also the technology, certainly. And I sincerely hope in the years to come or in the days to come, many of the youngsters who are very active in the field will take a lead to help our farming community, and we will be very, very happy to be associated with them, and we'll make it a reality for India to produce more fish and also earn double our farmers' income and also earn a good name for them. Okay, thank you.
Thank you very much, sir. For the information of the participants, Dr. Prabhudeva, sir, after his retirement, is coming out with his own tilapia hatchery very soon in the middle part of the Karnataka. And back, science is boring. I see Dr. Shiv Prakash, sir, though he is driving, is listening to us. Kindly park the vehicle to a side and recite a fisheries poem so that we can refresh our mind and go back with the great energy, sir. Dr. Shiv Prakash, sir, please. Unmute yourself, sir. Okay.
Okay.
Yes, sir.
Am
Yes, sir.
I audible?
Yes, sir.
Okay.
Yes, sir.
Okay, I was on the way to bring my wife from college. It's a very nice webinar, and world experts have come on the screen. Very pleasant to listen to all their expert advice. Okay, let me, Magada, I would like to focus on environment today.
Yes,
sir. We all need to have a clean air, clean atmosphere, clean, environment, so that people in the world live healthy. Now we have to overcome this COVID situation. So in order to have a healthy environment, we all need to care for our environment. So my 4 lines are on climate change. My poetry is on climate change. In order to make life on earth safer, we must set right our home on earth in order. Accelerate efforts to make it greener. Decelerate the mad rush to abuse nature.
Internet connection. Please come again.
Okay. In order to make life on earth safer, we must set right our home on earth in order. Accelerate efforts to make it greener.
Okay.
Decelerate the mad rush to abuse nature. Reduce greenhouse gas emissions to cool it further. Use renewable energy sources to prevent the imminent disaster. hold on to the traditional wisdom to make life happier, lest we might prove that we don't deserve to live on earth any longer. Thank you. Thank you very much.
Thank you very much, sir. Now we will take the question through mails. We have given the feedback link. Kindly share your feedback. Be critical. Don't use the feedback form to appreciate the resource person or the organizers. Send the critical questions so that we will come out with the standard operation or the standard package of practice for biofloc technology for different fish species. So now we are come to the end of the webinar. On behalf of Karnataka Veterinary Animal and Fisheries Sciences University, on behalf of all the participants, I thank our Vice Chancellor, Dr. H.D. Narayan Swamy sir, Dr. B.V. Shiv Prakash, Director of Research, Dr. N.A. Patil, Director of Extension, and all our mentors, Dr. C.K. Murthy, Dr. Seenappa, Dr. K.N. Prabhudeva, Dr. Y. Basavaraj, Dr. Vasudevappa, Dr. Senthil A. Dean, College of Fisheries. I thank all the dignitaries, all the resource persons, and you can see my team, Dr. Manjappa, Dr. Rajanna, Dr. Harsha. Please come on one screen, please. Stand behind me, please, and see my entire team. Close this, okay. Dr. Rajanna, Dr. Shruti, Adarsha, all the young team have joined together, and first time I see representation from 17 countries, and of late I received queries from many more countries, and it's a learning for us managing the system. That's why we have called the technician Mr. Deepak. He assisted us since morning, and there were few hiccups in the beginning. Definitely we will not repeat such mistakes in future. And next webinar, many people have requested us to organize a webinar on RAS. Very soon we will try to contact the right people, experts. Not that we are experts, we will provide the right platform to share their views. Definitely we will take the good and try to disseminate to the needy. And I thank all my university officers, my mentors, and all my colleagues. And if you are happy with our program, show a victory sign, raise your right hand or left hand, whatever it is. Show victory. Sir, victory sign. And Yoramji, victory sign. So thank you. Take this photograph, Dr. Harsha. All the participants, show your right hand with victory sign. And I take this opportunity to thank all the speakers, resource persons, and participants on behalf of my university, Karnataka Veterinary Animal and Fisheries Sciences University, and FRAC, Hebbal, FRAC Bangalore, and Dr. Lingdal has joined us from Marine Fisheries Research Information Center, Ankola, Dr. Vijay Kumar from Bhutnal, and many more people have worked. And I am little poor in system management. These people have helped me. Thank you very much. One more time, kindly raise your hands for a photograph. Victory sign, please. Babita, Eric. Thank you, Balu. Thank you very much. Thank you. And thanks, Professor Yoram. Namaste from India. Dr. Eric, namaste. What do you say in Israel, Professor Yoram? Salam? What do you say this? For thanks. Salam. Okay. Thank you, Eric. What do you say in Kenya for thanks? Namaste. Okay. I'll unmute Eric. Okay. Thank you, Dr. Debtano. Spread your wings to entire nation. Hitendra, I see under our bottom, Mr. Irendra is doing wonderful work. I don't know why he is doing silently. We want to take Hitendra to the entire world. You are doing wonderful work. we are with you. And Jagadish Ningappa, please join with Hitendra. Blend your RAS science with biofloc. And I see a lot of promising people help the Karnataka farmers to produce more fish and earn more money. Make business with the entrepreneurs, not with the farmers. And definitely I will make a big consortium as Yoram as our chief patron, and we will take the baton of success to the entire nation. Thank you very much, Shivprakash sir, C.K. Murthy sir, Prabhudev sir, Director Ramachari, Joint Director Mr. Dinesh Kaller, Sikvir Nayak sir, Deputy Joint Director, and all the fisheries officers in the entire state. I thank each one of you for joining this webinar and make this a grand success. Dr. Suprita, you want to say something finally?
Speakers, scientists, and more importantly, coordinators and participants. Thank you.
Thank you.
Thank you.
Professor Yoram, we will come back to you. There are so many questions. Okay.